Okay. Hey guys, waiting for the stream to go live. Just give me a second and then we can begin. Right. So I can see two people are live and the stream is also there. So we'll begin today's class. So before we begin today's class, uh, I want to show you guys what I have planned for today. Right. So first of all, I have a few MCQs. Uh, right. So we can attempt these MCQs at the end of today's class. And before that, I have your entire session planned out, right? And this session will be very useful for those of you who are appearing for your prof examinations, as well as it will help clear out concepts for those of you who have forgotten these, right? So we were getting some answers for the first question, right? So the answer given by, so since people have started answering it, I will discuss the first question. Question number 1. Right. So other people can also start answering. Initiation of impulse starts in. Where does initiation of impulse start? Right. So what is your answer, guys? Sangeet MK has answered this as D, and Anurag Joshi has answered this as B. Okay. Aishwarya S Kumar is also answering this as B. So this is a previous year question from Nimhans. Right. Now all the central institutes have a common entrance called as INICET. This is a very old question, but is still important. And like I told you guys, I'm just doing this MCQ because some people started answering it already. We'll first move to the theoretical part and then we'll be doing the MCQs. And you guys can hit the like button on this video if you guys like this video. And you can also join my Telegram channel, that is @Unacademy, NEET PG Chetanya, right? Where I'll update you about all my Unacademy sessions in a dedicated way. And you can also obtain the PDFs of these sessions, although they're also uploaded here on YouTube, right? All of you have answered B mostly, so that's correct. All of you have given the right answer. The initiation of impulse, it is starting in the axon hillock and the initial segment of the axon. So impulse here is the action potential, and that is where it begins. This is a straight line from your Genong's textbook of physiology. Okay, so before I begin today's class, I have to go through the subscription pitch. Let's do that, and let me tell you a little bit about myself. Okay. So welcome to Let's Crack NEET PG, the YouTube channel of Unacademy for the NEET PG division. About me, my name is Chetanya Mittal. I'm an MBBS at All India Institute of Medical Sciences, AIIMS, New Delhi, India's best medical college. All India rank 4 in the AIIMS exam, All India rank 5 in the JIPMER exam. I'm also a KVPY and NTSC scholar, right? So we're going to do the nerve muscle physiology today, and this is part of the first prof series that I'm doing, which is especially for the first year MBBS students, to show you guys that Unacademy has some really quality content, whether it be for your prof examinations or for your PG entrances, right? And you can see that we are going to begin in a minute. And yes, Anurag, bahut din baad class liya. Actually, I was on a trip. I was traveling this month, so because of that I could not take a class through most of the month, and I'll be taking it through these last 4 or 5 days. So I hope all of you have already downloaded the Unacademy learning app. If you haven't done that, please do that and use my referral code Chetanya10 to unlock your free plans. Okay? Unacademy is offering some of its courses, the live classes, some of the quizzes for free, and for that you will need a referral code. So use my referral code Chetanya10 and unlock the free plan on the Unacademy app. Okay? Here you have the top educators who are going to be teaching for Unacademy for NEET PG, and you can see a lot of them are from my institution, right? And we have special classes, that is free live classes here on Unacademy, where you will experience everything that is there on Plus absolutely free of cost. I have also made a few special classes for you. So this is my Unacademy profile. You can visit this link and you can see what are the classes that I have made. So these are all the free special classes that I have made. Recently, I did an MCQ marathon for abdomen on Unacademy. You can go and watch that. Then there are plus courses, which are logged courses. These are the ones that you get when you take a plus subscription. So I have also made a course on GI physiology where I've covered A to Z everything that you need for understanding gastrointestinal physiology. सर डाउनलोड किए हैं पर आपका आईडी नहीं दिखा रहा. ओके, मेरा आईडी नहीं दिखा रहा. ओके, मेरा आईडी नहीं दिखा रहा तो एक काम करना, वहां पे चैतन्या मित्तल सर्च करना. It will be there, or otherwise go to your mobile browser and just enter this. You will be able to access this link, or you can also access this from my Telegram channel, that is @Unacademy NEET PG Chetanya. ठीक है, ये Telegram channel का लिंक है. Okay, I'll put the link in the chat also. अच्छा, अब ये एक लिंक डाला है, इस पे क्लिक कर लेना. इट शुड कम टू योर प्रोफाइल. ठीक है, नाउ ट्राई टू सर्च इट. Right, so on Unacademy you have batches which are undergoing for syllabus completion. Intensive revision batch for NEET PG 21. Recently INI CET batch for 2021 has been started, so you guys can take your subscription very quickly and join all of these batches. So my referral code is Chetanya 10 and use this referral code to take whatever subscription you take. Wherever you have the option of entering a referral code, you can attempt free mock tests if you download the app. So you can see there are T20 mock test series going on. ठीक है, तो बहुत सारे टेस्ट चलते हैं. ये टेस्ट नंबर 106 चल रहा है अभी. एंड कॉम्प्रिहेंसिव सिलेबस कवरेज फॉर ऑल सब्जेक्ट्स, फिजियोलॉजी, ENT, बायोकेम, मेडिसिन, एवरीथिंग इज गोइंग ऑन. And you know why Unacademy is the easiest way to learn online? It provides you flexibility not only in terms of timetable but also in terms of language and in terms of unlimitedness. It means that you can re-watch your lectures, you can re-attempt all your tests, so it's really flexible. And you have the option to chat with your educator. So I've opened the YouTube live chat here on my mobile phone. So whatever you write, I can see, and if you have any doubts, you can keep letting me know. Interactive polls and the ability to learn wherever you want, whenever you want. So go ahead and install it. The other thing is there is an iconic subscription that is offered by Unacademy where you have not only the benefits of Unacademy Plus, but you also get complete access of Prep Ladder, complete access to Prep Ladder, including its video lectures, including the question bank, including rapid revision course, and including notes, right? So this is everything that you get in Unacademy Iconic, and this is everything you get in Unacademy Plus. So Unacademy Iconic, you can take the 1 year or the 2 year plans. The 1 year plan is for 55,000 and the 2 year plan is for 77,000. Just apply my referral code. My referral code is very simple. It is just my name and 10 after it, because you will get a straight away 10% discount. It can be availed on any course, whether it be for NEET PG, UPSC, NEET UG, IIT. Let me know whichever course you take it for, right? And you have the option of no cost EMI on all plans which are above 6 months. On all plans above 6 months, you have no cost EMI. So it's more beneficial to take these 1 year or 2 year plans. This is an Unacademy secret that the 1 year and the 2 year plans are priced the best. ठीक है, इनकी pricing सबसे अच्छी होती है. If you take the plus course, after applying my referral code, you will get it for 24,750 if you take the 1 year plan. And the 2 year plan, it will cost you 36,000, right? So do use my referral code if you take any subscription and try to take it as a new user, right? So this is where you can enter the referral code and this is where you can apply your credits. Credits are a very good way to get some additional discount, okay? And you should always do this on Android or on desktop, okay? Because on the mobile application, if you apply the referral code, you will not get a discount, you will get free weeks. Okay, so it depends on how you want to use it. If you want free weeks, take it on iOS. If you want an additional discount, take it on Android. Okay, so let's begin the class for physiology first prof essentials, right? So there was a PDF that I had sent on my group regarding all the important first prof questions, all the important first prof questions for physiology, right? So some of you said that you also want answers to these questions and notes for these questions. So this class is basically to discuss each and every topic, each and every question from those particular notes. You can obtain all of this here. These are the links where you can get the respective PDFs also for this. You can join these groups and you will be able to get the PDF for this particular session, right? So these are the questions that were there for nerve muscle physiology. These are all the questions that we are going to cover today. Nerve muscle physiology, right? So just have a look at these questions. We are going to do genesis of graded and action potentials, neurons and neuroglia, myasthenia gravis, its differentiation with Lambert-Eaton syndrome, genesis of resting membrane potential, right? And mechanism of contraction and relaxation in skeletal muscle and smooth muscle, including excitation, contraction, coupling, energy sources for skeletal muscle, saltatory conduction, neuromuscular junction and transmission, where we will also discuss neuromuscular blockers. And then I have some miscellaneous topics where I have discussed nerve injuries and types of nerve muscle fibers. Okay? So I have taken up everything that you need to do from this topic and I have presented it in the form of your prof friendly question answers. It is also again very useful for people who are appearing for NEET PG because I will be covering concepts. So ये सारे important main concepts हैं and इन्हीं में से question आने का chance है. And at the end I also have MCQs for you. So this is going to be a little long session. So gear up for it and I hope you all are ready for the session. Please write a yes if you all are ready for the session and we will begin with our first question. We will begin with our first topic. Just watch this entire session. Your nerve muscle physiology is prepared for your prof examination. You should have no worries in that. Okay? And before we begin, एक बार just like button दबा दो so that I know that you people are ready. You guys are ready for the session. Just hit the like button for this video. Okay. Sangeet has written yes. ऐश्वर्या का भी yes आ गया, अनुराग का भी yes आ गया. Okay, great. Okay. Lisha का भी yes आ गया, right? So if anywhere anything you don't understand, all you have to do is just write it in the chat box to explain it again. Anywhere anything that you don't understand, all you have to do is in the chat box write समझ नहीं आया. Or just write that you did not understand. I will repeat it in whatever language you want me to repeat it, in whatever detail you want me to repeat it. Okay? Chalo. Let's start with graded potential and action potential in case of nerve. The first question that was there in the questions was graded potential and action potential in case of nerve. So the first thing that we will do is we will differentiate between graded potential and action potential. So what is the difference between graded potential and action potential? The first very important difference is that graded potential is a change in membrane potential that is confined to a very small region. Alright, it is confined to a very small region. If this is your entire neuron, right? So graded potential will remain, let's say, limited in this area. So graded potentials, they are limited to small areas, changes in membrane potentials which are confined to relatively small regions, whereas action potentials, they spread from one place to another. So action potentials, they will cover much larger areas because they are going to spread over the membrane. So that's the first thing. That first difference is related to the area of spread. Okay. The second thing is that graded potentials are only localized membrane potential changes because they take a small regions. Okay. So they are not propagating, they are non-propagating, whereas action potentials, they propagate. That is, if an action potential, it begins somewhere here in the neuron, it will travel the entire length of the neuron, whereas a graded potential, it will begin here and it will end by here. Okay? So graded potentials, they are small changes, which are non-propagating, and action potentials, they are propagating. So that is, they move along the length of the nerve fiber. So second is propagation. Second difference is propagation, right? What is the third difference? The third difference is that graded potentials are produced spontaneously. The graded potentials, they can be produced in 2 or 3 ways. One of the ways of their production is that they are produced spontaneously by diastolic depolarizations, which are known as pacemaker potentials. They take place in your heart or in your GIT. Okay, so they can take place with the help of pacemaker potentials spontaneously, or they can be due to neurotransmitters, which are known as end plate potentials, which take place at the nerve muscle junction, NMJ, neuromuscular junction, or they can be due to some environmental mechanisms. For example, these are known as generator potentials or receptor potentials. For example, if I touch your nerve ending here, if I apply pressure here, or for example, if I do a pin prick here, there will be some graded potentials that are produced in the receptors here. Those will be known as receptor potentials, or they can be due to stretch, that is mechanosensitive. So there are multiple ways in which graded potentials can be produced. So based on the mechanism of production, there are multiple ways in which a graded potential can be produced, right? But an action potential, it is only produced by membrane depolarization. It is initiated by depolarization of the membrane, and it is only produced in the nerve, in the muscle, and in some glands. Whereas you can see graded potentials, they are also produced in receptors. They are also produced in receptors. They are produced in almost every cell. Graded potentials can be produced in almost every cell, but every cell cannot have an action potential, or every cell cannot generate action potentials. Action potentials can only propagate in cells which are properly excitable, which have the property of conduction. Only those cells can carry out action potentials. Okay, so that's a difference between that. These are the first 3 points of difference between graded potential and action potential. Okay, so you won't find all of these summarizations anywhere else. They are taken from multiple books. Okay, so I've compiled all these answers from you from 2 or 3 textbooks. I'll tell you: A.K. Jain, right? Genong's textbook of physiology, Guyton, and Byrne and Levy. So I've referred, and Bijlani also, right? So I've referred to these 5 textbooks to form these answers, and trust me, these answers are model answers. These answers are answers that will fetch you 10 out of 10 marks if you write them in your prof examinations. So just listen to everything that I'm saying, because that's definitely going to be beneficial for you, right? So we're done with this much. Let's move ahead and talk about other points of differences between graded potential and action potential. So first thing is that action potentials are all or none phenomena, right? Either the action potential will be 0 or it will be complete, 0 or complete. So that is all or none phenomena. Whereas if you talk about graded potentials, the word graded by itself tells you that they are graded responses. That is, it can be 0, it can be 0.2, it can be 0.4, it can be 0.6, it can be 0.8, it can be 1. So there is variability, and they can have summation. There can be summation of graded potentials. For example, if you have a graded potential of 0.2 and if you have a graded potential of 0.4 and they are given at the same time, they can summate to give you a graded potential of, let's say, 0.5. Okay, so there can be summation. There can be summation of graded potentials. There is temporal summation and spatial summation. Chhod do wo baat abhi ke liye. Right now, just focus on the fact that graded potentials are graded responses and action potentials are all or none phenomena. Okay. Next thing, action potentials are always depolarizing. Action potential अगर जा रहा है तो हमेशा depolarizing होगा. Action potentials are always depolarizing. Whereas graded potentials, they are basically changes in charge distribution. So they can be either depolarizing or they can be hyperpolarizing. For graded potentials, it's not necessary that they have to be depolarizing. They can either be depolarizing or they can be hyperpolarizing, right? That makes sense to you? They can either be depolarizing or hyperpolarizing. Then the next thing is that there is no specific threshold for graded potential. A graded potential can be very small also. A graded potential can be very large also. But for action potential, there is a certain threshold of depolarization that must be reached before an action potential will be carried out. And that threshold is given here, right? So there is a certain threshold for an action potential. Graded potentials, you do not have any refractory period. Therefore, summation is possible. Whereas action potentials, you will have refractory period. We will discuss in detail how an action potential is produced. and you will see what is absolute refractory period, what is relative refractory period, and what is the ionic basis of refractory periods. So this is a direct question that can come to you. Differentiate between graded potential and action potential in nerve fibers. This is everything that you have to explain. And we have also summarized for a conceptual understanding that what is the difference between a graded potential and an action potential. Okay? So are you guys satisfied with this particular answer? If you have to write this answer for, let's say, 10 marks, will you be able to write it? Will you be able to write this answer for 10 marks? We will continue. There are certain more points that are there, and we'll also talk about in detail about action potential and its genesis. So next thing that I'm telling you here is that graded potentials, they're very important in signaling over short distances. Okay? Because I told you they are present over a short area. They are present over a short area. Whereas action potentials, they are long distance signals of the nerves because they are going to propagate. So therefore they are important in long distance signaling. They're important in long distance signaling, whereas this particular one is important in short distance signaling. And it also decrements or it attenuates with distance. What does that mean? Graded potentials, they are not self-replicating. They cannot travel without decrement. So action potentials, so let's say there is a point A and there is a point B. Okay? If there is an action potential that has to go, it will continue with the same energy intensity. It will continue throughout with the same energy intensity. Whereas a graded potential, it will die out in a while. Graded potential will die out in a certain while. Okay? Graded potential, they are not going to travel without decrement. They will travel with attenuation. They will decrease with some distance. Okay? Okay, how do I show this to you in a better way? So if this is a graph of how a graded potential will be, okay, let's do it in the form of energy. Okay, so I said an action potential, the energy of an action potential with as long as how long it travels, it remains constant. Alright, it is traveling without decrease or decrement. Whereas a graded potential, the energy will decrease very steeply. Okay? In a short distance, a graded potential will get exhausted. It will decrement. There is also a length factor in which the graded potential becomes a certain fraction of its initial fraction. Okay? So let's not get into that. अगर physics में RC circuit वगैरह पढ़ा था, तो I hope you remember there was a constant called as tau. Do you remember this formula? Tau is equal to RC. You remember something like this? Right? So similar to that, here also you have a length factor or an attenuation factor in which the graded potential will decrease. But for now, try to remember that in certain distance there will be decrement in graded potential, whereas an action potential travels without decrement. Graded potentials are physical phenomena and action potentials are biological phenomena. Okay? So let's come to the genesis of graded potential. Right? See here, applications of sub-threshold stimuli do not produce an action potential because they only have an effect on membrane potential due to passive redistribution of charge. What does this statement mean? What I am saying is that if you apply a threshold, so there was a difference between graded potential and action potential, that graded potential, they do not have threshold, whereas action potentials, they require threshold. So if you apply a stimulus that is below threshold, you will produce a graded potential. And if you apply it above the threshold, you will produce an action potential. So for a sub-threshold stimulus, you will produce graded potential, which is basically due to passive redistribution of charge. Okay? Now, if it is towards the negative side, that is known as an electrotonic graded potential, and if it is towards the positive side, it is known as cat electrotonic graded potentials. Alright, so what is cat here and what is an here? This means anodic electrotonic potentials. Why are they called anodic? Because what does anode do and what does cathode do? These are cathodic electrotonic potentials. Yeah, so they are called as anodic and they are called as cathodic. You will tell me why they are called as anodic and why they are called as cathodic. Just think of it and you will be able to answer. So anodic electrotonic potentials, they are hyperpolarizing. They are hyperpolarizing, right? And cathodic ones, they are depolarizing. They are depolarizing. So they will produce a local response. They will produce a local response at a particular level where sodium voltage gated channels will open. And at that point, there will be a firing level. At that point, there will be a firing level. Okay? So what I am doing is, at every level, I am increasing the strength of the stimulus. At every level, I am increasing the strength of the stimulus. And when that stimulus reaches a threshold that is known as the firing level, the action potential will propagate. The action potential will propagate. So that is known as propagated. So a propagated graded potential is basically an action potential which will come at a threshold, and that threshold is known as the firing level. ठीक है, ये बात समझ आई? So action potential, they are also known as impulses. They are also known as spikes. They are transient. They are transient, self-regenerative, electrical, conducted without decrement or signal loss, and they are transfers of signals, right? So what are the various phases in the genesis of action potential? Just have a look at those phases. Okay. Initially, what is there? This is known as the resting level, which is the RMP. Okay. अनुराग जोशी को समझ नहीं आया. Okay. Let's begin from here again. So whenever I produce a graded potential, I can do this in two ways. Okay. This was your resting membrane potential. This was your resting membrane potential minus 70 millivolt. Right? Now I can do two things. I can either inject positive charge or I can either inject negative charge. That is, I can either take the membrane towards the negative side, that is towards, let's say, minus 90 millivolt, or I can either take it towards the positive side, that is, let's say, towards minus 35 millivolt. When I take it towards the negative side, I am producing certain graded potentials. I am producing certain graded potentials, and these are known as anelectrotonic potentials. When I take it towards the depolarizing side, I am again producing certain potentials. Okay, and if these are graded potentials, they will soon decrement, right? At a certain level, there will be a firing potential that is reached, and then you will have an action potential, and then you will have an action potential, right? And that level is known as the firing level, at which your depolarization reaches a threshold beyond which your sodium voltage gated channels are activated and they will produce a positive feedback in their own opening. ठीक है, अभी आते हैं उस बात पे. How is an action potential produced? I have not discussed how is an action potential produced. I have only discussed this graph for you, where you have to note what is a firing level, what are cat electrotonic potentials, what are anelectrotonic potentials. ठीक है, अभी ज़्यादा कुछ मेजर बात आई नहीं है. Just have a look at this graph and then you will understand it better. Okay. There is a resting level at which your cells are normally present at. There is a resting level at which cells are normally present at. That is known as resting membrane potential, right? You know the value. It's about minus 60 millivolt to minus 70 millivolt, and it is variable for various cells. What is the reason? What is the reason for RMP? Because potassium is more permeable, so its equilibrium potential, it dictates what the RMP is going to be. Okay? So the resting membrane potential, it is due to the permeability of potassium. It is due to the permeability of potassium. At this stage, sodium is almost impermeable to the cell membrane, but potassium is highly permeable. Potassium concentration inside the cell is more and potassium concentration outside is less. Okay? Potassium is the main intracellular cation. But inside you have lots of negatively charged proteins. And they are responsible for the negative RMP inside the cell. When you receive a potential here, when you receive, let's say, a graded potential here or stimulus, graded potential or stimulus, there will be passive depolarization, there will be redistribution of charge, and if that reaches this certain level known as a threshold, if that reaches a threshold, there will be opening of sodium VGC, there will be opening of sodium voltage gated channels at this threshold level. And when these open, there will be a steep rising phase of depolarization which will overshoot beyond 0 millivolt, and the cell will go to almost plus 20 millivolt. That is known as the peak of depolarization. That is the peak of depolarization, right? After the peak is reached, at some point, the sodium VGC, they get closed. Sodium VGCs, they get closed, and the potassium VGCs, they will open now. Potassium VGCs will open. Okay, so now potassium, it starts to go out of the cell. Potassium, it starts to, initially what had happened, this was the cell, there was entry of sodium, there was entry of sodium that caused depolarization. Then the sodium VGC closed and the potassium VGC opened, so there was exit of potassium from the cell. So that is now known as repolarization. That is now known as repolarization. So the cell moves to the original state, and because the potassium VGCs, they will take some time to close, so the cell goes even beyond the RMP, and that is known as after hyperpolarization, or after hyperpolarization is also known as, you can simply call it hyperpolarization or after hyperpolarization. And then slowly the sodium potassium pump will restore the gradients. The sodium potassium pump will restore the gradients. So whenever you are asked about phases of action potential or genesis of action potential, or phases of action potential, you basically have to draw this curve. You basically have to draw this curve and you have to explain all the phases. So what are the phases that you have to explain? The first phase is the resting membrane potential. At rest, the RMP is negative, potassium is more permeable, so its equilibrium potential, it will dictate the RMP. When you move further, there is a depolarized state in which the membrane becomes permeable to sodium ions because there is a passive redistribution of charge. The depolarization, it is due to the entry of sodium ions, and in large nerve fibers, right? In large nerve fibers, there is overshooting because of opening of sodium VGC, right? What was the overshoot phase? This overshoot was this one. It going above 0 millivolt, That is the overshoot phase. That is only there in large nerve fibers, right? And what was the repolarization state? Within few milliseconds, the sodium channels, the sodium VGCs, they begin to close. And the potassium channels, they will open. Then the rapid diffusion of potassium to the exterior re-establishes the normal negative resting membrane potential. And finally, the rectification of original ionic concentration. This is known as rectification. That is done by the sodium potassium ATPases. So again, to summarize this, what is happening? First, there is RMP due to potassium permeability. Then there is depolarization due to sodium entry. Depolarization is due to sodium entry. Then what happens? The sodium VGCs, they will open. That will cause depolarization. Then sodium VGCs get inactivated and potassium VGCs open. That will cause repolarization. And finally, the sodium potassium pump, it will cause rectification. That includes restoring the original ionic concentrations. So that's the simplification of everything that we have studied. Now there is one concept known as refractory period. So let's see what is refractory period. Okay. So this is the sodium channel. That is shown to you. In the resting state, what happens? The activation gate, there are two gates of the sodium channel. There is an activation gate and there is an inactivation gate. In the resting state, what happens is the activation gate is closed. closed, and the inactivation gate is open. The inactivation gate is open. When there is depolarization, that is, there is entry of sodium, and there is positive charge on the interior, on the cytoplasmic site. What happens? The activation gate, it opens. Okay? Activation gate opens. The inactivation gate, it has to get closed, but the inactivation gate is a little slower. So the inactivation gate takes some time to get closed. Okay? So this is opening and this is wanting to close, but it is very lazy. So it takes some time to close. And after some time, what happens? The activation gate is open, but the inactivation gate has closed. Right? And since the inactivation gate is closed, right? Since the inactivation gate is closed, what happens? There is refractory period. So even if you give membrane depolarization, even if you depolarize the membrane further, nothing can happen, because even if the activation gate is open, the inactivation gate has closed the sodium channel. Okay? In the resting state, the channel is closed because of closure of activation gate. In the activated state, the channel is open. Why? Because the inactivation gate is taking some time to close and the activation gate is open. And when you have depolarization, when depolarization is maintained, the inactivation gate closes. Then after repolarization, only after repolarization is complete and charges have been restored, the inactivation gate will open again and the activation gate will close. Only then the inactivation gate will open again. Right? You're clear? Are we clear? Okay, now there is a small thing. So this was about the absolute refractory period. This was the relative refractory period, right? So during the repolarization phase, during the entire repolarization and some part of after hypolarization, there is absolute refractory period. So absolute refractory period means कि जितना मर्जी और depolarize कर लो, there will not be a second action potential. Relative refractory period is the remaining part, this part here. Okay? This is the part where the inactivation gate has opened. Now. Now if you stimulate it, you will have to apply excess stimulus, but still there is a possibility that it can open. Once the inactivation gate has opened, you will require excess stimulus, that is known as relative refractory period. Okay? Relative refractory period means that it is refractory to normal stimulus, but if you give supramaximal stimulus, if you give a very high amount of stimulus, there will be a second action potential. So refractory period, it will determine the maximum possible frequency for firing of a neuron. It is the maximum, you will identify the maximal possible frequency of firing of a neuron, and you should remember this, that frequency of firing of action potentials, that is often used to encode certain information for certain signals. For example, in your inner ear, right? The frequency with which the neurons fire, they will often help you understand the various characteristics of the sound that is being received. They will help you identify the various characteristics of the sound that is being received. Okay? So we've covered this more or less in a very detailed way. Okay? So one more thing is the effect of calcium on sodium channels. Have you ever wondered that why does hypocalcemia cause tetany? Why does hypocalcemia cause tetany? Have you heard of parathyroid tetany? Parathyroid tetany. If you remove the parathyroid glands, the calcium levels in blood, they will fall. The calcium levels in blood, they will fall. So what calcium does is that calcium, it binds to the exterior surface of the sodium channel protein molecule. It binds to the exterior surface of the sodium channel, and it increases the threshold by altering the electrical state of the channel. So what does the calcium ion do? The calcium ion, its role is to increase the threshold of the sodium channel. What is the role of calcium channel? Let's write this down. Calcium, it will increase the threshold of activation of sodium channel. Now, if the calcium levels in the blood are low, what will happen? This threshold, it will decrease. That will lead to, if there is low calcium, the threshold is also low. Low calcium, low threshold. So, more excitability. Okay. So, the muscle will remain in a state of tetany. There will be continuous excitations. Okay. There will be continuous excitations. So I hope you have understood this also, and we'll move with the feedback during depolarization and repolarization. What is the positive feedback and what is the negative feedback? You also have to draw this during your examinations. What is the positive feedback during depolarization and what is the negative feedback during repolarization? Positive feedback during depolarization, if there is a depolarizing stimulus, that will open some sodium voltage gated channels that will cause increased permeability of sodium, that will lead to increased sodium flow into the cell, which will cause depolarization. This depolarization will further give feedback for opening of more sodium VGC. So this is a positive feedback cycle. So if sodium VGCs opens, there is more sodium entry, which causes more depolarization, which causes more opening of sodium VGCs. So that is the reason why action potential is so powerful. Why once the threshold is reached, why the action potential will always go to a all or none phenomena. Why it will always go to completion. Why it is so self-propagating. And the moment there is inactivation of sodium channels, this depolarization will stop. Okay. So depolarization also causes opening of potassium VGCs, but this is delayed as compared to with respect to sodium VGCs. ठीक है, same time पे नहीं होता है. Potassium VGCs are slower to open. Potassium VGCs are slower than sodium VGCs. ठीक है. So opening of potassium channels, that will cause increased permeability of potassium, and that increased permeability of potassium will cause increased flow of potassium out of the cell. That will cause repolarization of membrane potential, and it will lead to negative feedback, and it will cause repolarization of cell membrane. So it's a very powerful feedback mechanism that operates during the action potential. There is positive feedback during rapid depolarization and there is negative feedback during repolarization, and this positive feedback is responsible for the all or none phenomenon. Once the threshold is reached, this is responsible for the all or none phenomenon. Okay, so I hope you are clear with the first question. I hope we can move to the second question. ठीक है, कोई भी डाउट हो यहां तक तो प्लीज बता दो. Please tell me it's clear. Okay, great. ठीक है. The second part of this, the second question that we are going to do for today, that's really very easy. ठीक है, first question थोड़ा लंबा था because we are covering the basic fundamentals of everything. ठीक है, so the first question was a little lengthier than I expected. The second question, it's very simple. It's about neurons and neuroglia. ठीक है, basically you can refer to these notes anywhere you get confused. Neurons, they are 10% of all the cells. They are the basic structural unit. There is dendrites, axons, myelin sheath, synaptic terminals, cell body, that is soma, nucleus, nodes of Ranvier, missile granules, and neurofibrillae. Okay, so you've studied all these in your class 11. You've studied all these in your class 11. So it's very similar here regarding neurons. There isn't much that is new. You know about the structural classification also. What are apolar neurons? What are unipolar neurons? What are bipolar neurons? What are multipolar neurons? What is important to remember for you guys is pseudo-unipolar neurons. What are pseudo-unipolar neurons? Example, the neurons present in the dorsal root ganglion. Example, the neurons present in the dorsal root ganglion. Or those of the mesencephalic nucleus of trigeminal. Mesencephalic nucleus of trigeminal. So these are very important. Dorsal root ganglion and mesencephalic nucleus of trigeminal. It is the only pseudo-unipolar neuron of CNS. Only one in case of CNS. Dorsal root ganglion are pseudo-unipolar neurons in the peripheral nervous system, whereas the mesencephalic nucleus of trigeminal is a special case. It is a pseudo-unipolar neuron inside the CNS. It is a pseudo-unipolar neuron inside the CNS. But you know about the functional classification. Some of them are sensory neurons, some of them are motor neurons, and there are interneurons. Sensory neurons, they are afferent. These are motor neurons are efferent, and interneurons will lie between them. ठीक है, तो ये बहुत simple classification है. I hope you are able to do this structural and functional classification. We'll move to the important thing that for today, that is neuroglia. We'll move to neuroglia. So remember, there is one thing that you have to know, that neuroglia constitute 50% of volume of CNS. 50% of the volume of CNS. Okay, and they constitute over 90% of the cells in CNS. 90% of the cells in CNS. Okay, so if there are total of 1 billion neurons, if there are 1 billion neurons, I say if there are 1 billion neurons, there will be 10 billion, almost 10 billion neuroglia. Okay, and they will occupy 50% of all the space in the CNS. Okay. Anurag Joshi, yes, these are my handwritten notes which I have prepared for all of you for your prof examinations. ठीक है, specially बैठ के मैंने ये सारे आंसर लिखे हैं so that you guys can have notes of at least a few topics, and ये topics आपके जल्दी जल्दी हो जाए. ठीक है. Second thing, neuroglia, they are electrically coupled via gap junction and they proliferate lifelong. They are electrically coupled via gap junction and they proliferate lifelong. So classification of neuroglia, there is macroglia and there is microglia. There is macroglia means large glial cells, microglia means small glial cells. Macroglia will further include astrocytes, oligodendrocytes and Schwann cells and ependymal cells. ठीक है, इसमें astrocytes are fibrous astrocytes and protoplasmic astrocytes. Fibrous astrocytes are found in white matter and protoplasmic astrocytes are found in grey matter. ठीक है, that's about this much. क्या उसके अलावा there are some special radial cells, special glial cells. ठीक है, for example there are something known as Muller cells. They are found in retina. Muller cells, they are found in retina. They are special glial cells of the retina. ठीक है, Bergman glia cells are glial cells of cerebellum. Then there are enteric glial cells in gut wall, and there are satellite cells which are the glial cells of the autonomic ganglia. They are the glial cells of the autonomic ganglia. ठीक है, and embryologically there were some glial cells called radial glial cells. ठीक है, the only important one that I find here is the Muller cells and the Bergman cells. ठीक है, ये दोनों याद रख लो. Satellite cell people usually remember, so these three you can remember and rest it's okay. ठीक है, this much is fine. Hello, let's move ahead from the classification and let's move to the functions of astrocytes. ठीक है, this is a usually asked as a 10-mark question. What are the function of glial cells? ठीक है, 10 मार्क का क्वेश्चन आ जाता है function of glial cells पे. and you should be able to write something more than support. ठीक है, because everyone knows that the function of glial cell is support, function of astrocyte is blood brain barrier, function of microglia is phagocytosis. ठीक है, उसके beyond you should be able to write some points, because you are writing your answers of first year MBBS, not class 11 class 12. ठीक है, so your answers should have something extra from what you learned in class 11 and class 12 that will show in your marks. ठीक है, so functions of astrocytes are star shaped cells which provide support. They form the blood brain barrier. Absolutely true, these two things, they are the main functions of astrocytes. Besides that, astrocytes, they help in mopping up of leaked ions. Right? Then they help in enhancing synapse formation, and this strengthens synaptic transmission. There is something known as tripartite synapse. Tripartite synapse क्या हुआ? ठीक है, पति पत्नी और वो, what is पति पत्नी और वो? Tripartite synapse. What do I mean here? There is a synapses between neuron 1 and neuron 2, but क्या होता है, इन न्यूरॉन्स के बीच में एक सेल आ जाता है जिसे कहते हैं ग्लायल सेल. ठीक है, this is pati, this is patni, and this is wo, pati patni or wo, tripartite synapse. It means that the glial cell, it helps in enhancing synapse formation and it strengthens the synaptic transmission. At this synapse, it will perform some other functions like taking up and degrading released neurotransmitters. It will take up and degrade released neurotransmitters. ठीक है, then it also plays some role with the potassium levels here. It forms neural scar tissue. It releases neurotrophic growth factors. ठीक है, what are the neurotrophic growth factors? Basic fibroblast growth factor. There are a lot of growth factors that are released here. They are also released by astrocytes. So remember, secretion of neurotrophic factors. Then it provides metabolic support. It provides certain nutrients like lactate. ठीक है, then they are responsible for reuptake of glutamate, which is a neurotransmitter. ठीक है, so they prevent glutamate excitotoxicity. What is glutamate excitotoxicity? If extracellular glutamate levels rise, the neurons, they will undergo degradation. Why? Because there is excess calcium entry via NMDA receptors of glutamate. There are some receptors known as NMDA receptors, right? And they are sensitive to glutamate. So if there is excess glutamate, these NMDA receptors will cause excess calcium entry, and calcium is a signal for apoptosis. And that is the cellular basis of glutamate excitotoxicity. Besides that, astrocytes help in reuptake of potassium also. They regulate neural networks. ठीक है, general सी बात है. So these are all the points that you have to write when you talk about what are the functions of astrocytes. So there are a good 10 functions that you can write for astrocyte. ठीक है, 10 function होता है astrocyte के. So you should be able to write at least 6 to 7 of these functions. Only then you will get a decent number of marks. ठीक है. Besides that, functions of microglia, they are basically spider-like phagocytes. So they will dispose of debris, they will mediate immune responses, injury, infection, seizure, everything. And they're also APCs. What are APCs? Antigen presenting cells. So they are basically modified macrophages of the CNS. Antigen presenting cells bhi hote hain ye. Like all other macrophages, they are also antigen presenting cells, right? What about ependymal cells? Ependymal cells, they will line the cavities of the brain. What are the cavities of the brain? Ventricles. And what are the cavities of the spinal cord? They will also line those. Okay. Then they will secrete CSF. Second, the epidermal cells are neural stem cells. They can regenerate to form other neurons, and they can also regenerate to form other glial cells. Please remember, the stem cells of the CNS, they are ependymal cells. Besides that, you have oligodendrocytes in the CNS for myelin production, Schwann cells in the PNS for myelin production, support and insulation. Satellite cells provide support in autonomic and sensory ganglions, right? So we are done with the second question. Okay guys, clear so far? Now we will cover 2 questions together. In fact, we will cover 3 questions together. Okay, question C and question number F are the questions from the first slide that I had shown you that we are covering right now. ठीक है. So you can get a question to write a note on myasthenia gravis. You can get a question to write a note on Lambert-Eaton myasthenic syndrome, right? Or you can get a question to differentiate between these two. You can get a question to differentiate between myasthenia gravis and Lambert-Eaton syndrome. ठीक है. तो in any case all you have to do is write the points that are relevant. ठीक है. तो मैंने differentiation तो दिया हुआ है. Okay, and if you have to, let's say you get an answer question to write a note on myasthenia gravis, just write the left side of this differentiation, and if you get to write a note on Lambert-Eaton myasthenic syndrome, just write the right side of this question. ठीक है. Very quickly let's do this as well, so that we are done with almost 40% of our topic. I'll just go and get some water for myself. You guys can also take a 2-minute break. Give me 2 minutes. Right. So meanwhile, you guys can and try to plan out in nervous system. So that's why I'm doing nerve muscle first. Then I'll also try to do topics from the nervous system, that is thalamus, cerebellum, etc. Okay. So you can tell me freely topics that you want to understand. Should we start with myasthenia gravis versus Lambert-Eaton syndrome? Okay. So first of all, before I begin, the main difference that you should know is that myasthenia gravis, it is a defect of which channel, and Lambert-Eaton syndrome is a defect of which channel. So tell me, myasthenia gravis is a defect of what channel, and Lambert-Eaton syndrome is a defect of what channel? I need this answer from you guys. Myasthenia gravis क्यों होता है और Lambert-Eaton myasthenic syndrome क्यों होता है? CVS कर लेंगे, ठीक है? CVS, a lot of people have said. We'll try to do CVS also. Okay. Tell me, why do you have myasthenia gravis, and tell me, why do you have Lambert-Eaton myasthenic syndrome? अनुराग जोशी, जसविंदर कौर, बताओ क्यों होता है myasthenia gravis? Okay, so these are very useless differences. ठीक है, this is the incidence of occurrence of myasthenia gravis and Lambert-Eaton myasthenic syndrome. This is very rare. Okay, and this is slightly more common, but this is also pretty rare. Okay, but you will still see myasthenia gravis patients, because I have seen myasthenia gravis patient in AIIMS, and it's not very difficult to identify cases of myasthenia gravis. Okay, right. So myasthenia gravis is a autoimmune disorder due to formation of antibodies against acetylcholine receptors. ठीक है, acetylcholine receptors के against. And Lambert-Eaton myasthenic syndrome, it is due to blockage of which channels? Due to some channels. ठीक है? कौन से channels है? I'll tell you. They are basically presynaptic calcium channels. Presynaptic calcium channels. Myasthenia gravis, it can occur in any age, and it is more common in the 20s for women, and it is more common in the 60s for men. ठीक है? So it is called as bimodal distribution. ठीक है? अगर 20 साल की औरत है तो उसमें myasthenia gravis ज्यादा कॉमन है, 60 साल का बुढ़ा है तो उसमें myasthenia gravis ज्यादा कॉमन है. That these are the main ages in which myasthenia gravis will occur. Whereas Lambert-Eaton myasthenic syndrome, it is usually adult onset, and it is similar in case of men and women. There is no differentiation for the age, right? So here the antibodies are formed against nicotinic acetylcholine receptors, like antibodies against nicotinic acetylcholine receptors, right? And here the antibodies are formed against calcium VGCs. Calcium VGCs. Calcium VGCs are presynaptic calcium VGCs. They are responsible for release of neurotransmitter. Okay. Myasthenia gravis, it mainly affects extraocular muscles, your muscles of the eye. So there is drooping of the eyelid. That's one of the most common symptoms of myasthenia gravis. Okay. There are 2 or 3 types of myasthenia gravis. In one of them, mainly only eyelids are affected. There are other types in which other muscles are also affected. So antibodies against ACH receptors are produced, and the main muscle that is usually involved are extraocular muscles, which will cause droopy eyelids. Whereas in case of Lambert-Eaton syndrome, generally proximal muscles of the lower limb are involved. What are the proximal muscles of the lower limb? Basically your thigh muscles, right? The muscles which are proximal. Okay? So they are the ones that are involved, and because of that you will have waddling gait. Okay? Waddling gait ho sakti hai, and difficulty in raising your arms. So proximal muscles of the upper limb can also be there, because of which you will have difficulty in raising your arms. Okay? That is what will happen in limbs. The very important clinical difference, very important clinical difference. This was your very important physiological difference. The very important clinical difference is that in myasthenia gravis, the patient gets tired after repetition. Why? Because if you repeatedly stimulate the number of quanta of neurotransmitter that is released is decreased. The number of quanta of neurotransmitter that is released gets decreased because acetylcholine gets exhausted, ACH gets exhausted. Whereas in case of LEMS, after repetitive stimulation, calcium, it will accumulate in the presynaptic terminal. Okay? And there will be improvement. Okay? Try and understand this last point. I hope you were able to understand it. This is your presynaptic terminal. Okay? यहाँ से release होना है neurotransmitter to go here on the postsynaptic terminal, right? So what is going to happen is, here what is there in case of myasthenia gravis, ACH receptors, they are non-functional. So if you again repeatedly stimulate it, what will happen? ACH that is released, it will get used up because ACH synthesis takes time. Okay? So if ACH is released in excess due to repeated stimulation, soon ACH here will finish. ACH gets exhausted. So there is tiring after repetition. In case of myasthenia gravis. Okay. But what happens in case of Lambert-Eaton myasthenic syndrome? The release of ACH requires the entry of calcium. Okay. So in that, this channel is blocked. Now if I repeatedly stimulate it, okay, let's say in some time some small amount of calcium was able to go in, then on the second stimulation some small amount of calcium was able to go in, third time some small amount of calcium was able to go in, fourth time another small amount of calcium was able to go in. So finally this will be able to cause the release of ACH. Okay? So it will improve upon repetition. That's one of the main differences that you have to understand between myasthenia gravis and Lambert-Eaton myasthenic syndrome. If it is asked in your viva, the first 2 points that you should mention, the first points that you should mention before you mention anything else is that there is first tell about what receptors or what channels are damaged. Second thing you should tell about is what happens after repetition, what happens after repetitive stimulation. This will worsen, this will improve. Okay, so that differentiation is very important. Okay, besides that, there is a correlation here with rheumatoid arthritis, systemic lupus erythematosus, and polymyositis. These are all autoimmune disorders. So myasthenia gravis is also an autoimmune disorder, and it is well correlated with other autoimmune diseases, other diseases of autoimmunity. And there is believed to be a role of thymus. Why? Because helper T cells are sensitized against thymic protein. So because of these, there is believed to be a role of thymus, hyperactive thymus. So one of the treatments here is said that you can remove the thymus gland. One of the treatments that is said for myasthenia gravis is removal of thymus, but the main treatment is acetylcholine esterase inhibitors, ACHE inhibitors. You might have heard of this. What happens in case of the neuromuscular junction? What is done is, let's take a black screen. If this is your neuromuscular junction, once acetylcholine is released, here acetylcholine molecules reach the synaptic cleft. There is an enzyme that acts here called as acetylcholine esterase, which will break acetylcholine into acetyl-CoA and choline, right? So we will use inhibitors of acetylcholine esterase, which will increase the levels of acetylcholine in the synaptic cleft, and they will help to alleviate the symptoms of myasthenia gravis. Okay. The very important point of correlation with regards to Lambert-Eaton myasthenic syndrome, which is often asked in quizzes. Okay, quiz का MCQ वगैरह होता है. So it is often mentioned that the patient has small cell lung carcinoma, because 40% of patients who have Lambert-Eaton myasthenic syndrome also have small cell lung carcinoma, and there is also a reason for that. It is said that similar antibodies are produced to attack cancer cells. So the antibodies that are produced by the body to attack these cancer cells, they also bear resemblance to presynaptic calcium channels, and that is the reason for Lambert-Eaton myasthenic syndrome in patients who have small cell lung carcinoma, right? Then aminoglycoside antibodies also produce similar syndrome. Here, acetylcholine esterase inhibitors are almost useless, and the main drug here that is used is aminopyridine, which are presynaptic calcium channel blockers. So they will block the presynaptic potassium channels, which are responsible for repolarization. So they will maintain depolarization for a long time, and they will help the calcium VGCs to release calcium, causing release of neurotransmitter. ठीक है, समझ आया ये वाला बात? What does aminopyridine do in case of Lambert-Eaton myasthenic syndrome? Here again I will draw the cleft for you. Here you had calcium channels. Okay, these calcium channels, they are responsible for efflux of potassium causing repolarization. They are responsible for repolarization, and your calcium channels here, they are not working well. The calcium channels here, they are not working well, calcium channels, and they require depolarization because they are calcium VGC. So if you block these potassium channels, you block them using aminopyridine, there will be sustained depolarization. Okay, that will help in calcium influx. Causing release of neurotransmitter. Clear? Will you be able to write a 10-mark question for the differences between myasthenia gravis and Lambert-Eaton myasthenic syndrome now? Will you be able to write a 10-mark answer for this differentiation? Okay, so this answer was written purely from Ganong's textbook of physiology. Yeah. So believe this answer. This is a very authentic answer. This is a complete answer, and this is an ideal answer. Okay. Do you guys have any doubts so far? Were you able to understand everything? If you're liking the video so far, you can hit the like button. Are there any doubts, or are you guys clear with everything? Everything clear? Guys, can I move to the next question? Hello, guys. I can see a lot of people are watching. Guys, try to be more interactive, try to be more involved with the chat, try to ask your doubts, try to give me feedback, try to tell me uh, if there's anything that I should repeat. Okay, I'm not getting any response. I'll move ahead. So now the topic we are going to cover is going to be mechanism of contraction. Mechanism of contraction. Yeah. So first we'll do skeletal muscle. First we will do in case of skeletal muscle. Okay, thank you, Bhupinder Singh. And then we will do in case of smooth muscle. Then we will do in case of smooth muscle, so that we can also compare the differences between these two. Yeah. And what we are going to do is called as excitation contraction coupling. For skeletal muscle, we'll discuss something known as excitation contraction coupling. Okay. So what do you understand by coupling? What do you understand by coupling? When two things are done together. Okay. There is something known as excitation. There is something known as excitation. And that excitation, it produces contraction. And how does excitation produce contraction? That is known as excitation contraction coupling. That is known as excitation contraction coupling, right? So mechanism of contraction and relaxation in skeletal muscles. Let's start with this. So if there is a muscle fiber, if there is a muscle fiber and there is an action potential in that muscle fiber, right? After some time, what happens? There is an increase in the calcium levels of that particular muscle cell. There is an increase in that in the calcium levels of that cell, and that brings about the contraction of that muscle, right? So this was excitation, this was contraction. And what do you think is the mediator between this excitation and contraction? That is the increase of myoplasmic calcium levels. That is the increase of myoplasmic calcium levels. That is the mediator between the excitation and contraction. As a whole, this thing is known as excitation-contraction coupling. So what is the process of excitation? Excitation is the transmission of impulse along the neuromuscular junction, genesis of muscle action potential that spreads along the sarcoma. That is the not sarcoma here, sarcolemma, right? That is the plasma membrane of the muscle cell, right? And what is contraction? It is a shortening of length of the muscle or increase in tension of the muscle. Okay, so there is a coupling between these two phenomena. There is a coupling between the excitation phenomena and the contraction phenomena. In skeletal muscle, the source of calcium is intracellular. So where do you get this myoplasmic calcium? You don't get this from the outside, but you get this from the sarcoplasmic reticulum. You get this from the sarcoplasmic reticulum. So the source of calcium is intracellular. Source of calcium is intracellular. So that's the first thing that you have to know here regarding the calcium of skeletal muscle. Okay. So next we will see how there is going to be this coupling. First we have to know something about triads. Right? What are triads? They are present at A-I junctions. What are A and I junctions? It means there is an A band and there is an I band in your sarcomere. Remember A band and I band, you did this in class 11th. A band and I band. So at the junction of that A band and I band, there is something known as triads. So what are triads? Basically, there are some invaginations of plasma membranes, and these invaginations are known as T-tubules. These invaginations, they are known as T-tubules. Okay, around these T-tubules, there are certain cisterns. There are certain extensions of sarcoplasmic reticulum, and they are known as terminal cisterns. Okay, these extensions of sarcoplasmic reticulum are known as terminal cisterns. And together these three they are known as triads. Okay, so what is a triad made up of? A triad is made up of T-tubule plus two terminal cisterns. Then these terminal cisterns, they continue in the form of L-tubules. Okay, clear? What are triads? Okay, it's very important to understand what are triads to understand excitation contraction coupling. Okay, so what happens is there is an action potential that comes, and that action potential causes release of calcium from sarcoplasmic reticulum. Now how does that happen? Now how is there release of calcium in response to an action potential? So the action potential, it comes inside the T-tubule. It comes here, right? So an action potential, it was on the surface of the sarcolemma. Then it comes inside into the T-tubule. Inside the T-tubule, there must be some voltage sensor. That senses that an action potential has arrived. So there is a voltage sensor in the T-tubule. Membrane. Now there is something for the release of calcium. So these are called as calcium release channels. They are present in the terminal cistern membrane. There are calcium release channels in the terminal cistern membrane. So somehow this voltage sensor must be coupled to this calcium release channel. This terminal voltage sensor in the T-tubule, it must be coupled to the calcium release channel in the terminal cistern. Now have a look. There are certain things known as foot processes in the gap between T-tubules and terminal cisterns, right? There are certain things that are known as foot processes. So they are going to mechanically directly connect two channels. Those channels are known as ryanodine receptor and the dihydropyridine receptor molecules. Okay, this RYR channel, there is something known as DHPR, okay, which is the voltage sensor. and there is something known as IRYR, which is the ryanodine calcium release channel. Okay, and there is a mechanical interaction between these two. Okay, there are 4 DHPR molecules and they are directly attached via foot processes to a channel known as RYR, which is a homotetramer, and it has certain foot processes. A muscle action potential, it propagates into the T tubules, and there is a voltage sensor that is called as DHPR, dihydropyridine receptor molecule. Okay, this molecule is, so this is DHPR. It is directly attached. Mechanical interaction is present between the DHPR and the ryanodine receptor. RYR stands for ryanodine. So there is a coupling between DHPR and ryanodine receptor, which is a calcium release channel, and there is a mechanical interaction between these two, which causes the release of calcium, and it further causes contraction. Okay, this much is clear to you. How there is coupling at DHPR and RYR levels. Okay, so your class 11th and 12th knowledge does not give you knowledge of DHPR and RYR. Okay, so you have to be very careful and you have to note these down very carefully. What is DHPR, what is RYR, and what is the coupling between these two molecules, right? Clear? Percentage force, calcium. There's a graph between percentage force and calcium. So as calcium levels increases, the tension in the muscles, it increases. Okay. Hi Uday Sharma. In case you guys like this video, please do hit the like button on this video. I would really appreciate it. Thank you so much. Yeah. So now once you understand this, now you will be able to understand contraction in case of skeletal muscle. Now you can understand contraction in skeletal muscle once you have understood excitation, contraction, coupling. So what is happening? There is increase in levels of calcium. Now what happens when the calcium levels increase? That will be explained by the sliding filament model, which was given by Huxley and Huxley for skeletal muscle. There is something known as sliding filament model. You studied this in your class 11th, but we'll do it in greater detail. Okay, we'll do it in greater detail. So first tell me, do you guys have any doubts so far? Do you guys have any doubts so far? Do you have any doubts? No. There is a T tubule that refers to transverse tubule, and there is a terminal cistern. There is nothing known as terminal tubule. Okay, see here, DHPR, it is present on T tubule, and ryanodine receptor calcium release channels, they are present on sarcoplasmic reticulum terminal cisterns. Okay, this should be clear to you. Okay, thank you for the doubt. थैंक यू फॉर द डाउट भूपेंद्र सिंह. समझ आ गया भूपेंद्र सिंह क्या बात करी अभी तक? चलें आगे, स्लाइडिंग फिलामेंट मॉडल पे चलें। Okay, chalo. Let's start with the sliding filament model, which was given by Huxley and Huxley. There is something known as troponin C. There is something known as troponin C. What is troponin C? Troponin C is a calcium sensor. Troponin C is a calcium sensor. What does troponin C do is, troponin, it binds calcium, alright? The myoplasmic calcium that increased in the sarcoplasm, that increased in the cytoplasm of the skeletal muscle cell, it senses the calcium, right? It senses the calcium. Then it binds the calcium and it undergoes conformational change. It binds the calcium and it undergoes some conformational change. So what does it do is that, if you remember from your class 11, there was something known as tropomyosin filaments. There was something known as tropomyosin filaments which were covering the myosin binding sites on actin. They were covering the myosin binding sites on actin. So the troponin C, when it undergoes conformational change with the help of other troponin subunits, it causes unmasking of the myosin binding sites due to flipping of those troposin myofilaments, right? And then it opens these cross bridge binding sites. Because of that, you will have cross bridge cycling. You will have what is known as cross bridge cycling. Just have a look at what is cross bridge cycling. Okay? Just have a look at this animation and try to understand what happens. See, this was your myosin thick filament. This was your myosin thick filament. This was your myosin thick filament, and this was your actin binding head, and this was your myosin binding site. This got exposed when tropomyosin was removed due to the presence of calcium. This was exposed when the tropomyosin was removed due to the presence of calcium, which was sensed by TNC, right? So the moment this site gets exposed, right, this myosin binding head, it will go and bind with it. It will go and bind with it like this. Alright, it has gone up and bound with the myosin binding site. Then what happens is, there is something known as power stroke. It is also called as ratchet action. Okay, so what happens here is, this head, it will bend here and it will move here, right? This is called as ratchet action. Okay, so you can understand how it is pulling the thin filament on the thick filament. So the thin filament is sliding on the thick filament. Therefore, this is known as sliding filament model. Right? Then what happens? Once that is done, ATP comes and binds to it. Okay. Because of this binding of ATP, there is a conformational change and this tension is released. Okay. And this myosin can leave the actin binding site. There is dissociation of myosin from the actin binding site due to binding of ATP. There is dissociation. Okay, so have you heard of something called as rigor mortis? Have you heard of something called as rigor mortis. Have you heard of rigor mortis? Have you heard of rigor mortis? After death, there is no ATP. There is no ATP. Okay. So what happens? There is no ATP. There is no replenishment of ATP. So the myosin head remains in this bound state. It remains in this bound state. It cannot move until new ATP binds. It cannot it cannot get released. So it remains like this. Because ATP is required for what is known as recocking. Right? So for that you need hydrolysis of ATP. So ATP is not bound, so that is the basis for rigor mortis. And after that, this will get released. It will come down. ATP is present here. Again, there will be hydrolysis of ATP, and again the myosin head will be available for binding. This will now again bind, and then there will be again contraction. So you can see in this way, this filament will move. Okay. So you can understand how exactly is this working. Again, there is a power stroke. This filament moves. There is recocking, and there is again cross-bridge cycling. So see here from this point onwards, let's have a look at this entire animation again. To understand the cycling, it's must for you to understand this entire process. Okay. So just watch this very carefully. 1, 2, 3, 4, 5, 6, 7, 8, 2. Okay. So you understand the rhythm that is going for cross-bridge cycling. Right? You should be able to understand cross-bridge cycling with the help of this diagram. Now, when you have to draw it in your examination, you have to draw it in the form of a cycle. So what all will you write in that case? You will write the binding of the unmasking of calcium, binding sites due to increase in calcium. Then this will attach there. There will be ratchet action which pulls the actin filaments. After that, there will be binding of ATP. And once that ATP is bound, it is hydrolyzed, and that energy is used for recocking. And then again this goes and binds. So you understand what is recocking. So this was the cogged state. ठीक है, जब ऐसे होता है. And when ATP is hydrolyzed, this becomes like this. That is recocking. Okay. So please understand what is recocking, what is ratchet action. So these are a few terms that are related here to cross-bridge cycling. Those are the terms that you have to understand. Otherwise, this is pretty much very similar to the NCERT diagram that you might have done during your preparation for NEET UG. Alright, that you might have done during your NEET UG preparation. So I hope you understand what is cross-bridge cycling in muscle contraction now. You understand that? Yeah. So this is what is cross-bridge cycling. There is attachment, there is force generation, there is detachment, and then there is detachment. Okay. So that is the entire process that is going. And asynchronously, there are lots of cycling cross bridges, and they will ensure that there is continuous shortening or force generation. Okay. You will think that once that it gets detached, the muscle will go back. That's not true. Why? Because there are multiple muscle fibers that are undergoing cross-bridge cycling simultaneously. So together they will bring about shortening or force generation of muscle. Together they will bring that about. And there is amplification here. So there are hundreds of myosin heads. They will all contract together. They will bring about the contraction of a thick filament. Then there are hundreds of thick and thin filaments, and hundreds of thick and thin filaments will form sarcomeres. There are multiple sarcomeres in a myofibril, and there are multiple myofibrils in a muscle fiber. So there is an amplification that takes place at every step. Okay? What I have shown you right now in that animation is the contraction of a single myosin head, the ratchet action of a single myosin head. Hundreds of those will be occurring at the same time in a single thick filament. Hundreds of thick and thin filaments will be there in a single sarcomere. Then there will be hundreds and thousands of sarcomeres in a single myofibril, and that will get amplified because there are thousands of myofibrils in a single muscle fiber. Then there are multiple muscle fibers in a single muscle. So there is multiple levels of amplification that takes place from this small contraction, you produce actual visible muscle contraction, right? Got it? Now, quickly we'll talk about relaxation. Now, relaxation is an active process, and as you can understand, for relaxation, you will have to decrease the calcium levels. You will have to decrease the calcium levels. And to decrease the calcium levels, you will have to send back the calcium where it came from. Where did it come from? Sarcoplasmic reticulum. And to send it back, you will require some channels, and there will be active reuptake of calcium that is done through a channel known as sarcoplasmic smooth endoplasmic reticulum calcium ATPase. ठीक है, SERCA, that is known as SERCA, that is smooth endoplasmic reticulum calcium ATPase. And inside you will have certain calcium binding proteins that will hold the calcium with them to prevent leakage of calcium. Such proteins are known as calreticulin, calsequestrin, calbindin, and they are present inside the sarcoplasmic reticulum lumen. They will reduce the free calcium and increasing the uptake capacity. Okay, so you understand how relaxation takes place, and now these electrical signals will get coupled to so now basically what happened in the entire process, there was coupling of electrical signals at the sarcolemmal surface to intracellular calcium release, resulting in muscular contraction. That is a one line summary of EC coupling, one line summary of EC coupling. Okay, is there any doubt regarding the process of hand contraction in where trained out चलो कोई रिस्पॉन्स नहीं आ रहा तो आई मूव ऑन टू मैकेनिज्म ऑफ कॉन्ट्रैक्शन ऑफ स्मूथ मसल. लेट्स मूव टू मैकेनिज्म ऑफ कॉन्ट्रैक्शन ऑफ स्मूथ मसल. ठीक है. So first thing that you need to know is that what are the differences between smooth muscle and skeletal muscle when it comes to such contraction. Okay. First thing is that in case of smooth muscle cells, they are together structurally and functionally coupled. Okay. They will contract generally as a unit and they will always try to do their contractions in a synchronized way. Right? Structural coupling is through adherence junctions and functional coupling is through gap junctions. Right? Second thing, in case of skeletal muscle, you had sarcomeres. Which were having Z discs. ठीक है. तो Z disc आती थी, फिर उसके बीच में sarcomere present होता था, I band, A band वगैरह होता था बीच में, right? So that was a sarcomere where you had Z disc. Instead of Z disc, in case of smooth muscle, you have dense bodies. In case of smooth muscle, you have dense bodies and plaques, right? That's one thing. Then next, if I talk about thin filaments, here the thin filaments in case of smooth muscle, they are made up of actin plus tropomyosin, but they are devoid of troponin and nebulin. Okay. Remember, in case of skeletal muscle, you had something called as troponin, but here you do not have troponin. Here you only have actin and tropomyosin. Then you have caveolae, which are longitudinal invaginations rich in ion channels, receptors and transducers, and along with the sarcoplasmic reticulum, it regulates the calcium concentration. So here, from the extracellular fluid also, you are taking up calcium. You are taking up calcium from caveolae. You are taking up calcium from the extracellular fluid. So source of calcium is both ECF and ICF, right? Now there is phasic smooth muscle contractions. There are phasic smooth muscle contractions, right? And there are tonic smooth muscle contractions. Phasic smooth muscle contractions take place in case of intestine, and tonic smooth muscle contractions are present in the vascular system and the airways. Okay. What do you mean by phasic smooth muscle contractions and tonic smooth muscle contractions? Okay. देखो, phasic means there is a phase of contraction and then there is relaxation for some time. Then there is again some contraction and then there is relaxation for some time. Okay, so there are certain phases in which contraction is occurring. Tonic means there is a maintained tone of contraction. There is a maintained tone of contraction. The tone may increase, the tone may decrease, the tone may remain normal. Okay, so that is a tonic contraction. That is a tonic contraction. Okay, there is always some vascular smooth muscle tone if you talk about a blood vessel. Okay, because if that tone gets lost, there will be absolute vasodilation. Okay, so there is always some tone that is maintained in your airways and in your vascular columns, that is your blood vessels. Whereas in case of intestine, for example, if you take your peristalsis, there is contraction, then there is relaxation. There is contraction, there is relaxation. There is contraction, there is relaxation. There is contraction, there is a relaxation. So there is some phasic nature of that contraction. Whereas in case of vessels and airways, you have tonicity. You have tonicity, right? Then the electrophysiology of smooth muscle is also a little different as compared to the typical action potential curve that you have for a skeletal muscle. जो ये वहां पे तुम सीधा सीधा ये बना रहे हो, ठीक है, उसकी जगह there is a different varied electrophysiology. So there can be spikes, there can be plateaus, or there can be slow waves. When you talk about your electrophysiology, there can be these three kinds of things that can be there. So plateau, you can see, you can observe this in case of the ventricular muscle fibers. In case of ventricular muscle fibers, right? You can observe this there in case of your heart. ठीक है, तो उससे just compare कर रहा हूँ. And in case of spike potentials, you can see these in your SA nodes or your AV nodes. You can see spike potentials. And then there are slow waves which are seen in case of GIT. Slow waves, they are seen in case of GIT. Okay, so there is a wave, then there is some spike that comes, then there is again some downfall, then there is again a wave, there is a downfall. Okay, that is what happens in case of the electrophysiology of smooth muscle. So cyclical changes in calcium chloride and potassium conductances keep occurring in case of slow waves. In case of smooth muscle, remember, in case of skeletal muscle depolarization, it was due to sodium. In case of skeletal muscle depolarization, it was due to a sodium influx. It was due to sodium influx. In this case, the depolarization is due to opening of L-type calcium channels. The depolarization is due to opening of L-type calcium channels rather than due to sodium. Here, depolarization is due to opening of L-type calcium channels. These are slow to open and these are slow to close, right? Repolarization is due to potassium VGCs, or it can also be due to calcium activated potassium channels. So here the repolarization can also be due to calcium activated potassium channels. Another feature here that you saw for the smooth muscle is that the resting membrane potential of the smooth muscle, it fluctuates. As you see here, in case of slow waves, the RMP, it is fluctuating. The RMP is fluctuating, right? Now, when you talk about coupling here, there is electromechanical coupling and there is pharmacomechanical coupling. Okay? So, excitation contraction coupling here, EC coupling here, it is of two different types. It can be due to electrical activity or it can be due to some ligand, that is known as pharmacomechanical coupling. Pharmacological agent, basically a ligand, basically due to some ligand. And it can also be due to electrical activity, where it will be known as electromechanical coupling, Example for electromechanical coupling is in the intestine, you have slow waves which are superimposed with spike potentials. I have taught this in a great detail in my class on GI physiology, right? Yeah. So slow waves with superimposed spike potentials, they are responsible for contraction in case of intestine. They're responsible for contraction in case of intestine. So there is increased calcium entry. There is increased. Okay, wait. Sorry, my stream got closed. So there is increased calcium entry and then there is force generation. In case of vascular smooth muscle, there is some myogenic tone, and that is because of graded changes in membrane potential, which alter the calcium influx through calcium voltage gated channels. And there is some gradation in force. So what is this basically meaning? There is certain stepped means graded. Okay? Thoda thoda karke. There are graded changes in membrane potential. They will alter how much calcium comes in through calcium voltage gated channels, and there will be some gradation in the force. For example, if let's say 0.5 millimole calcium comes in, there might be a change in 0.5 let's say 0.5 Newton of force, for example, right? And then let's say 0.1 millimolar calcium comes in, the gradation may be 0.1 Newton force. Okay? So that might occur in case of the vascular smooth muscle, that is known as myogenic tone. So in both these cases, the important thing is that the change is due to some change in membrane potential. The contraction, the entry of calcium is due to change in membrane potential. So this is known as electromechanical coupling. Whereas when you talk about pharmacomechanical coupling, what happens is the contraction and the relaxation is in response to agents that do not produce any change in membrane potential. Okay, what is happening? For example, certain hormones, certain drugs, they have effects on vascular smooth muscle. They will act directly through, let's say, G protein coupled receptors, and they will bring about changes in calcium levels. But they will not change membrane potential. They will directly increase the calcium levels and they will cause smooth muscle contraction. So that is pharmacomechanical coupling. So there is electromechanical coupling and there is pharmacomechanical coupling. In skeletal muscle, you only had electromechanical coupling. You did not have pharmacomechanical coupling. So pharmacomechanical coupling is unique to smooth muscle. This is unique to smooth muscle. Okay, please keep that in mind. What are the other factors that you see here? One here, that there's also a step of non-cycling cross bridge formation. There is a step of non-cycling cross bridge formation. We'll see what this means. We'll see what this particular thing means. Second thing, this is thick filament regulated. The contraction here is thick filament regulated. Whereas if you see in case of skeletal muscle, what was happening? The tropomyosin was covering the myosin binding sites on the thin filament, and the thin filament was the actual place where there was regulation of contraction. It was deciding whether contraction will take place or not. It depended on whether the actin had exposed myosin binding sites. So contraction in skeletal muscle was thin filament regulated. In case of smooth muscle, it was thick filament regulated. It is thick filament regulated. Please keep that difference in mind. The next thing, in case of skeletal muscle, what was the calcium sensor? Troponin C was the calcium sensor in case of skeletal muscle. In case of smooth muscle, calmodulin is the calcium sensor. Okay. So very important, these 3 points regarding the differences between skeletal muscle and smooth muscle contraction. So you might also get this as an answer. So please remember to write these 2 points. Are we clear so far? Guys, any doubts so far? Once I move to explaining this, then it is another 5 or 10 minutes that I'll take to explain this. So before that, if you have any doubts so far, you can ask them, because this is a little difficult to understand. This is slightly more difficult than the skeletal muscle. So try to pay attention now. Please ask your doubts if you have any. Hello, we'll move to the next part, that is regarding the smooth muscle contraction. So I told you that the sensor here is calmodulin. The sensor here is calmodulin, right? Now calmodulin is a calcium sensor, right? So when calcium is there, calmodulin, it gets activated. Calmodulin, it gets activated. What it does is it goes and activates myosin light chain kinases. These are called as myosin light chain kinases. So they will phosphorylate the light chains of myosin light chain kinases. So when the calcium is increased, the calmodulin, it will form a calcium calmodulin myosin light chain kinase complex. And this is the active form of this kinase. What this kinase does is it uses ATP to phosphorylate the myosin. So there is phosphorylation of myosin, and that will activate myosin. So now you can see here, why am I saying that contraction is thick filament regulated? Contraction is thick filament regulated. Now you can understand why I am saying that contraction is thick filament regulated. Okay, because there is phosphorylation of this myosin which will activate this, and that will cause cross bridge cycling. That will activate this and cause cross bridge cycling. So cross bridge cycling, it is initiated by the phosphorylation of regulatory light chains of the myosin molecule by the myosin light chain kinase, which is activated by calmodulin bound with calcium. So what is happening exactly, if I draw it in a flow chart? Calcium comes, it binds with calmodulin, then it forms calmodulin, calcium calmodulin, and calcium calmodulin, it will activate myosin light chain kinase. It will form a calcium calmodulin myosin light chain kinase. This kinase is active. This will phosphorylate myosin light chains. These are regulatory molecules. They will get phosphorylated with the help of ATP. We will phosphorylate them. Okay, we will use a phosphorus. Once these chains, they are phosphorylated, they will take part in cross bridge cycling. Clear so far? What is happening? What is the role of calmodulin in contraction? What is the role of calmodulin in muscle contraction? Smooth muscle, right? Role of calmodulin in smooth muscle contraction. So you can have a look at this particular diagram. You can see here, this is the non-cycling part and there is a cycling part of cross bridge cycling. So what happens here? When myosin light chain kinase is active, it will activate the myosin. Here, this myosin will then go and bind to the thin filament. It will bind to the thin filament. Once it binds to the thin filament, what happens? There is contraction, that is known as power stroke. Then what will happen? There will be attachment of another ATP molecule that will cause detachment of this. Then if this remains activated, then this process keeps on repeating. Right? Now, how is contraction stopped here? How does contraction stop here? There is something known as myosin phosphatase. Now, myosin phosphatase, it will remove this phosphate group from myosin light chain kinase. It will remove this phosphate group. And once these light chains become dephosphorylated, they become inactivated. Once they become dephosphorylated, they become inactivated. Okay, so what you have to understand here is that contraction is dependent on calcium levels and it is regulated by myosin light chain kinases with the help of calmodulin. Okay, phosphorylation of myosin, that increases the myosin ATPase activity and that increases the affinity of myosin cross bridges. Here, in case of excitation contraction, the coupling can be delayed as long as 500 milliseconds, because you can see that there are so many extra steps that have to be carried out. There has to be phosphorylation that has to be done. There has to be so much that has to be done before an excitation can be translated to a contraction. So the coupling can be delayed as long as 500 milliseconds. That's almost 0.5 seconds. seconds, right? So if this much is clear, we'll move ahead. So remember one thing, that the myosin isoform in case of smooth muscle, it has slower ATPase activity. That is, there is 10 times slower cross-bridge cycling in case of the myosin isoform of the smooth muscle. Yeah, keep that in mind. Velocity of muscle contraction, it depends on the myosin ATPase. Velocity of muscle contraction, depends on myosin ATPase. Always remember that. Okay. The cross bridge cycling, it depends on phosphorylation status of the cross bridges. That is, how much is the activity of myosin light chain kinase divided by how much is the activity of myosin phosphatase. Cross bridge cycling will depend on how many myosin are active and how many myosin are non-cycling and how many are cycling. And that will depend on the ratio of these two enzymes, calcium calmodulin myosin light chain kinase versus myosin phosphatase. It will be defined by these two parameters. Is that clear? So far, is this clear? This was the main important understanding point when it comes to smooth muscle contraction. If you understand this, you will understand everything. You will be able to write everything. You will be able to understand everything, right? Now here, if I ask you a question here, if I dephosphorylate, just look at this. Don't look here, don't look here, don't look here, don't look here, don't look anywhere else. Just look at this particular step. Here ATP is bound to this, right? Now what if I dephosphorylate? What if I attach myosin phosphatase here? What happens? Okay, now it goes into a non-cycling state, right? So what will happen? In one way, इसका मतलब क्या हुआ? कुंडी लग गई. Kundi is equal to, do you know what is kundi in Hindi? It's the thing that you use to close a door. Alright, so this is known as latch state. Okay, so what will happen? The myosin, it will be in a contracted state, but it will not be cycling. So it's not using energy, it does not use energy, but there is tension. Right? So this is known as latch state. It's essentially equal to rigor mortis. Essentially, you can compare it with rigor mortis. Don't equate it to rigor mortis, but it is somewhat similar to rigor mortis. It is somewhat similar to what is happening in rigor mortis. What is happening? You are dephosphorylating the myosin light chain kinase. You are basically dephosphorylating the regulatory myosin light chains when the cross bridge is attached. And that is known as latch state, which is a low state of low cross bridge cycling, and there is a high tension and low energy state. So you are not using any energy, but you are still maintaining tension. And this occurs when there is dephosphorylation of myosin, when it is bound to actin and it retains the cross bridge in the force generating conformation, and it slows down the detachment rate. So what does this accomplish? This accomplishes a tone of contraction without spending energy. So for example, if I talk about your vascular smooth muscle, it has to remain in a contracted state. So either you spend energy in every cross bridge cycling, that would be very expensive. That is what you would have to do in case of a skeletal muscle. But in the smooth muscle, you can maintain it in a latch state. So you are not spending much energy, but you are maintaining tension. So that is the benefit of smooth muscle. That is the benefit of the step of myosin light chain kinase and myosin phosphatase. Now you will understand why that extra step exists in case of smooth muscle, because you have to maintain a latch state, Okay, do you understand this much? Have you understood what is the latch state? Have you understood what is the latch state? Upender Singh, are you still watching? Any doubts anyone has? We do an MCQ before we move ahead. Okay. Can you guys answer this? Okay, so Jaspinder is here. Should we move back to the theory or should we do this MCQ? चलो वापस थ्योरी पे चल रहा हम करते हैं इसको जल्दी से. Let's talk about electromechanical coupling and pharmacomechanical coupling. When you are talking about calcium, how does calcium entry take place? How does calcium exit? How does calcium entry take place? How does calcium exit? Right? So we are going to talk about this. So very simply said, calcium entry, there can be direct entry or the entry can be due to a second messenger. If there is entry due to second messenger, that is pharmacomechanical coupling. Okay? Then calcium exit, we'll also see how calcium exit takes place, right? Calcium exit is generally through what is known as calcium ATPase, right? Or it can also be due to a sodium calcium exchanger, which is a secondary active transport deriving its energy from sodium potassium ATPase, right? So let's do this again. Try to understand one thing. That calcium for smooth muscle, it can come either from the sarcoplasmic reticulum or it can come from ECF or it can come directly from ECF. Okay. So if I have to cause entry of calcium from ECF, there are some leaky channels that help calcium to enter. There are certain voltage gated channels. There are certain ligand gated channels and there are certain stretch activated channels. All of these help in the entry of calcium. There are also store operated calcium channels, SOCC, right? So all of these help to enter calcium inside. Now, if the calcium has to be released from sarcoplasmic reticulum, there are calcium induced calcium release channels, okay? Because this calcium will enter from here and that will induce further release of calcium from the sarcoplasmic reticulum. So there are multiple mechanisms by which calcium can be released electromechanically. Pharmacomechanically, via second messenger, there is a pathway known as IP3DAG, right? Through this mechanism, IP3 helps to release calcium. IP3 helps to bring about the release of calcium from the sarcoplasmic reticulum. So these are all the mechanisms in which calcium can be released, right? Now what about the exit of calcium? The exit of calcium, the calcium can go back either into the sarcoplasmic reticulum or it can go to the ECF, right? If it has to go inside to the sarcoplasmic reticulum, there will be a calcium ATPase that is attached here. And there is another calcium ATPase that can send the calcium out from inside the cell outward, or it can be done by a calcium sodium exchanger, and this sodium gradient, the gradient of sodium, it is maintained with the help of sodium potassium ATPase. So this is a secondary active transport, this is a secondary active transport, and this is a primary active transport. Okay. Hello. If I move ahead, what are the unique changes when it comes to a smooth muscle? What are the unique changes when it comes to a smooth muscle? First, graded changes are possible. Okay. You can have graded changes in membrane potential. You can have graded changes in myoplasmic calcium. You can have graded changes in case of force, which is very important for vascular smooth muscle tone, which is very important for vascular smooth muscle tone. Then second, alteration of calcium sensitivity of contractile apparatus is through kinases, and changing the myosin light chain kinase to phosphatase ratio activity is independent of calcium, right? So alteration of calcium sensitivity can be done of the contractile apparatus by changing the activities of the kinases or the phosphatases, right? Third, the relaxation is not entirely driven by fallen calcium. You can maintain tone, you can maintain latch state. Even if the calcium level is less, even if the calcium level is fallen, you can still maintain the latch state by dephosphorylating it, right? And the fourth unique feature is the feature of pharmacomechanical coupling. Then I've told you other unique features. You can write that in case of skeletal muscle, the calcium sensor is troponin C. In case of smooth muscle, the calcium sensor is calmodulin. Similarly, you can expand this particular answer. Okay. Now we are coming to something that is very important. What are the energy sources for a skeletal muscle? What are the sources of energy for a skeletal muscle? Okay. So what is the order in which various molecules are utilized by a skeletal muscle for its meet its own energy needs? What are the energy sources for a skeletal muscle? Okay. Going to discuss this now. You guys have any doubts? You go and get some water. Meanwhile, you guys can take a 2-minute break. I actually do need some water. Meanwhile, you guys can read the slide. Let's talk about energy sources in case of skeletal muscle. So you might have read that there is a particular order in which energy will be used up for a skeletal muscle. Okay. So the first source that the skeletal muscle uses for its energy is stored ATP. The first source of energy for a skeletal muscle is stored ATP, right? And that is exhausted in a few twitches. That gets exhausted in a few twitches. Within a few twitches, that energy of stored ATP gets exhausted, right? And it keeps getting replenished. It is replenished continuously. That's the first source. And how long does it last? Only a few twitches. I can say for a few seconds it will last. Not even few seconds, barely 1 or 2 seconds, right? After that, the next source that the muscle turns to is called as creatine phosphate. The next source of energy for a muscle is creatine phosphate. There is rapid replenishment of ATP via a single enzyme pathway. Now, why is creatine phosphate important? Because it can produce ATP from a single enzyme pathway as compared to other steps where you will require 5 to 10 enzymes. So what it basically does is you will have production of ATP, you will use creatine phosphate and you will make creatine. This phosphate is used to form ATP, right? So you will have replenishment of ATP, and creatine phosphate is going to last 5 times more than the ATP pool. It is 5 times more than the ATP pool, and it will last for a few seconds. Let's say this lasts for around 1 to 10 seconds. This will last for 5 to 10 seconds, right? This is going to last for a few seconds, creatine phosphate. You can see here myosin ATPase, it will help in contraction, calcium ATPase, it will help in relaxation, but ATP is continuously being used, whether it be for contraction, whether it be for relaxation, right? Then we will talk about the next source of ATP. Next source of ATP is glucose. Next source of energy is glucose, right? How is glucose being produced? Glucose is coming from the muscle glycogen and the blood glucose. And the muscle, it can easily work on glucose for 30 minutes. It can easily work on glucose for 30 minutes. These are all multi-enzyme pathways which are relatively slower. You will have glycolysis and you will have oxidative phosphorylation. You will have glycolysis and you will have oxidative phosphorylation. And most of the ATP here, most of the ATP is coming from oxidative phosphorylation at moderate activity levels. But as you increase the activity, as you increase the intensity of muscular activity, the contribution of glycolysis, that is anaerobic respiration, it will increase. Anaerobic contribution will be more if you increase the intensity of muscular activity. Then the contribution of anaerobic respiration becomes more. Then the intensity of muscular activity is increased. The contribution of glycolysis also increases. Then next we'll talk about fatty acids and triglycerides. Fatty acids and triglycerides, right? In this case, fatty acids and triglycerides, and there are also some amount of amino acids. They are used at the end, and they will take part when there is prolonged exercise. That is from few minutes to a few hours. Here, the muscle cells have some fatty acids. It can also be from the blood, and here you will produce energy using beta oxidation. Here you will produce energy using beta oxidation. So this is the long-term source of energy. When you have to exercise for a few hours, you will also turn to fatty acids and triglycerols for your exercise. So I hope you're clear with this much as far as this entire topic is concerned. Should we move ahead? Should we move ahead? Let's talk next about oxygen debt. Let's talk about oxygen debt. Oxygen debt is the oxygen that you have to use after exercise is over. Oxygen debt is the oxygen that you have to use after exercise is over, and that is used to replenish ATP and creatine phosphate and to clear out the lactic acid that has been produced. So for lactic acid metabolism it is used, and to replace the levels of myoglobin, replace the oxygen that was attached to myoglobin, right? And that is what oxygen debt is used for in case of muscle cell, right? Now we will move to another very important question that is related to genesis of resting membrane potential and its maintenance. Genesis of resting membrane potential and its maintenance. So in case of skeletal muscle, cardiac cell and neuron, the resting membrane potential is between minus 60 millivolt to minus 90 millivolt, right? In case of smooth muscle cell, it is minus 55 millivolt, and in case of RBC, it is minus 9 millivolt. Okay. So let's see here what's going to happen here. What's going to happen here is that first of all, you need to understand what is resting membrane potential. Why is there something called as resting membrane potential? There is one very common misconception. There is one very common misconception between even medical school students that if I am saying that the resting membrane potential is minus 60 millivolt, it means that there is negative charge on this side and there is positive charge on this side. They understand that there is net positive charge here and there is net negative charge here. That's not the case. That's not the case. Both sides are equally having equal amounts of positive and negative charge. Both sides have equal amounts of positive and negative charge. Okay, both sides have equal amount of positive and negative charge. The only thing in case of resting membrane potential is that there is separation of charges. All right, at the membrane, at the place where there is separation of the membrane, there is positive charge is excess near the membrane and negative charge is excess near the membrane. Okay, overall there is an equal amount of positive and negative charge. Overall there is an equal amount of positive and negative charge, but there is a separation of charges near the membrane. So it's somewhat like a dipole. It's somewhat like a dipole. All right. And if there is a reduction of this charge at the membrane, right, or a reversal of this charge separation, that is called as depolarization. And if there is an increase in the charge separation, that is called as hyperpolarization. So I hope you understand what is polarization. Polarization means separation of charges, formation of dipole. What is dipole? पॉजिटिव एंड नेगेटिव चार्जेस के बीच का ये जो एक पोटेंशियल जनरेट हो रहा है, दैट इज द polarized state, right? And if you decrease this polarization, that is depolarization. If you increase this polarization, that is hyperpolarization. So the resting membrane potential, it is due to separation of charges across the cell membrane, right? Now genesis of RMP. When we talk about genesis of RMP, the resting membrane potential, it is contributed by multiple factors. All right. There is an electrochemical energy difference. There is an electrochemical potential energy difference, and there is a so basically electrochemical means there is a chemical potential difference that is due to chemical gradient. That is due to chemical gradient, concentration gradient, and there is a electrical potential difference that is due to charge gradient. That is due to charge gradient, right? At equilibrium, the electrochemical potential must be zero. At equilibrium, the electrochemical potential must be zero. So 0 is equal to, this is the chemical, this is the electrical, right? So they are equal, we'll equate them, and you'll arrive to the fact that the potential is equal to this. This is equal to the potential, right? if you have to find the electrical potential or the chemical potential, it is given by this particular equation. So for example, if you are asked for a particular driving force, so if you are asked for the electrical potential difference due to a particular ion, you will use the formula minus 60 divided by Zx, where Z is basically nothing but the valency. Z is nothing but the valency. For example, for sodium ion, it will be 1. For calcium ion, it will be 2. For aluminium ion, it will be 3. Right? Then log of its concentration gradient. X inside, X outside. Right? That's what you have to do when you are writing the driving force. When you are writing the not the driving force, the electrochemical gradient. When you're writing either the electrical gradient or the chemical gradient, and they are equal when you talk about the equilibrium, right? So the resting membrane potential, it is given by an equation known as GHK equation, GHK, Goldman-Hodgkin-Katz current equation, right? Where the resting membrane potential is equal to 61.5 millivolt multiplied by logarithm of 10 into potassium outside divided by potassium inside plus alpha sodium outside plus alpha sodium inside. Now, what is this alpha? Alpha is the ratio of permeability of potassium and sodium. Ratio of permeability of sodium and potassium. Sorry, ratio of permeability of sodium and potassium. And since you know that permeability of potassium is very greater than permeability of sodium, alpha is almost equal to 0.01. Alpha is almost equal to 0.01. And if I take alpha to be 0, then you will realize that the resting membrane potential depends almost solely on potassium. But there is some contribution of sodium as well, right? So we will just discuss that in detail as given in your guidance. This was a complicated understanding of RMP. We'll do a simplistic thing. So when you write this answer, you will write for RMP, two conditions must be met. First of all, there must be an unequal distribution of ions. There must be a concentration gradient. First thing. Second condition, the membranes must be permeable to these ions. Alpha should not equal to be 0. Alright, only then can you consider that ion for maintaining resting membrane potential. Right? So directly if you see, there is a contribution of potassium, there is a contribution of sodium, and there is a contribution of the electrogenic nature of the sodium potassium pump. Why? Because it takes in 3 sodiums and sends out 2 potassiums, right? So if the contribution was solely due to potassium, the equilibrium potential would have been minus 94 millivolt. The RMP would have been minus 94 millivolt. Alright. So potassium has the maximum contribution because it is the ion to which the membrane is maximally permeable. Here you will have the contribution of the Gibbs-Donan phenomenon because the protein and ions are bound by the potassium and they are impermeable. So they are responsible for the negativity that is present inside. Then the sodium has an equilibrium potential of which is positive, which is towards the positive side. So it will take the RMP a little towards the positive side, but since it is less permeable, it cannot take it too far ahead. So it brings the RMP towards the positive side, that is from minus 94 millivolt, it brings it to minus 86 millivolt. Then the sodium potassium pump has further contribution of minus 4 millivolt and it brings the RMP finally to minus 90 millivolt. This will vary for different cells. This is for a typical cell that we are studying. Indirectly, the maintenance of ionic gradients is done by the sodium potassium pump. So sodium potassium pump has very little direct role, but it has a very major indirect role. So when you write genesis of RMP, please remember to write these 4 points. Genesis of RMP, when you write genesis of RMP, please remember to write these 4 points. 1, 2, 3, 4. Okay. Please do remember to write all of these 4 points. Next, if I talk about the contribution of potassium diffusion potential, take a contribution of potassium diffusion potential. So first we'll calculate what is the potassium diffusion potential. So first thing that you should know is that concentration of potassium inside is almost 140, and outside is almost 4. So 140 divided by 4 is 35. So the nurse potential here will be minus 94 millivolt. How does this come? Minus 61.5 into log of 35 by 1. That comes out to be minus 94 millivolt, right? And that is the contribution of potassium. That is how the contribution of potassium is calculated. Contribution of sodium is calculated by its ionic concentrations. Sodium inside to sodium outside is approximately 1:10, and the equilibrium potential of sodium is approximately plus 61 millivolt. But its contribution is lesser because its alpha is 0.01. So contribution of sodium is minus 86 millivolt. Contribution of sodium is minus 86 millivolt. It brings it from minus 94 millivolt to minus 86 millivolt. Alright. If I move ahead, contribution of the sodium potassium pump. It sends in 3 sodium, takes out 2 potassium. So its extra contribution is minus 4 millivolt. That will bring the net RMP to minus 90 millivolt. And then there is the electrogenic nature of the sodium potassium pump. So that's about the genesis of RMP. With that, we are done with the genesis of RMP. That is basically what you have to write when you write the genesis of RMP. Okay. So last 2 questions are left. After that, I have some miscellaneous things that I have to explain and some MCQs for you guys. So do stay tuned for the rest of the session also. You have any doubts so far? You have any doubts so far? Let's talk about saltatory conduction now. Let's talk about what is saltatory conduction, what are the implications of saltatory conduction, how does saltatory conduction work. So first thing that you should know is that saltatory conduction, it occurs in myelinated fibers, since it is conduction basically from one node of Ranvier to another node of Ranvier to node of Ranvier. That's what is saltatory conduction. Saltatory conduction, it increases the length constant and effective range of how much transfer is there of suprathreshold depolarization. And there is a high density of sodium and potassium channels at nodes, and they regenerate the action potentials only at nodes. So you are reducing the energy cost. So what are you doing? You are making the transmission faster. And you are also reducing simultaneously the energy cost. How do you reduce the energy cost? Because now you have to regenerate action potential only at the nodes instead of doing it throughout the membrane, right? So action potentials, they occur only at nodes of Ranvier, since myelin does not allow passage of ions, and this node to node conduction, this jumping conduction, is called as saltatory conduction. Current flows through the surrounding ECF outside the myelin sheath as well as through the axoplasm inside the axon, exciting successive nodes. Okay. Hi Sukanya. Okay. So the importance here is the importance of saltatory conduction. What is the importance of saltatory conduction? Because of these jumping intervals, because the conduction is in quick jumps, right? Rather than going like this, the velocity is increased almost 5 to 50 folds, right? The velocity is increased almost 5 to 50 volts. and it saves energy. It saves approximately 100 times less loss of iron. So it saves approximately 100 times energy. It provides excellent insulation with the help of myelin. So there is a 5,000 times increase in resistance, and there is a decrease in capacitance, about 50 times decrease in capacitance. So there is repolarization occurs with little transfer of ions. So you don't have to transfer much ions. So you save lot of energy. All right. That's about this particular answer. Now after that, I'll move to neuromuscular junction and transmission at the NMJ. I'll also discuss the diseases of the NMJ and the various drugs that act on the NMJ. ठीक है, तो ये last question है. It's a little lengthy question as compared to the previous one. The previous one was a short question. So we'll simply do this part, and after that, like I told you, I have some additional topics that I have to teach you: classification of nerve fibers, then a differentiation between type 1 and type 2 fibers, and classification of peripheral nerve injuries. So once we are done with that, we'll move to MCQs, and that will be the end of this lecture. So we'll try to complete this lecture in another half an hour. Okay. So if you guys have any doubts so far, you can let me know. Otherwise, I'll move to neuromuscular junction and transmission. If you have any doubts, please let me know. Are there any doubts? Okay, right, Sukanya. So we can move ahead. Great. So let's talk about neuromuscular junction and transmission now. So first of all, you have to know that what is the need for transmission. The need for transmission is that a synapse is having a great distance. The distance between a presynaptic and postsynaptic terminal is very much, right? You cannot have direct flow of ions like this, or you cannot have direct flow of electricity like this, right? So the need for transmission is that the large gap, it will attenuate transmembrane potential difference by 105 times, right? So therefore you need to have some signal that goes from this end to this end, that carries the signal from this end to this end. Okay, so that is the need for neuromuscular junctions and transmission. If you look at the nerve and the muscle, so there is a lot of difference between them. There is a lot of difference in this neuromuscular junction, in the difference in terms of the length. There's a length difference here, right? There's lots of space between the nerve fiber ending and the sarcoplasm, right? So that has to be bridged by some transmission. A motor neuron and muscle fibers it innervates are together referred to as motor unit. So sometimes you are asked to define motor unit. Okay? A lot of times you will see the question, define motor unit. So you should know the definition of motor unit. Now when we talk about the structure of the nerve muscle junction, or a structure of the neuromuscular junction, structure of NMJ, this is what do you see? You see that there is a presynaptic terminal, which is basically the nerve fiber, and there is a muscular part. So in the nerve part, there is lots of mitochondria because you require ATP here, There are synaptic vesicles here which are filled with neurotransmitters acetylcholine. There is a presynaptic membrane, there is a synaptic cleft, right? And there is a postsynaptic membrane. Okay? There is an active zone where there is lots of release of this neurotransmitter, and there are calcium channels which cause the influx of calcium, and that calcium influx will cause neurotransmitter release. There is a basal lamina, there is a junctional fold. They will increase the surface area. Then there are voltage gated sodium channels that are present on the postsynaptic terminal. There are voltage gated sodium channels that are present here, and there are acetylcholine receptor channels that are present here, right, on the junctional folds. So they will be the ones that will respond to acetylcholine. These are nicotinic acetylcholine receptor channels. Okay. If I move ahead, what are presynaptic events? There is entry of sodium ion. Why? Because there is conduction of action potential. That will cause the opening of calcium voltage gated channel. That will cause the entry of calcium inside the cell. That will cause calcium entry. And calcium entry will cause opening of potassium voltage gated channels. That will cause repolarization, and that repolarization will close the calcium voltage gated channels, right? So that is what happens on calcium entry. Okay. You have to understand this in a more detailed way. There are two things that are happening together. There is entry of sodium ions. Entry of sodium ions causes opening of calcium voltage gated channels. That will cause entry of calcium. Entry of calcium will do two things. First, it will cause fusion of acetylcholine vesicles with the membrane and will cause exocytosis and quantal release of acetylcholine. What is quantal release? It means that acetylcholine is released in a graded way. One synaptic vesicle has a specific number of acetylcholine neurotransmitters which will produce a specified response at the postsynaptic terminal, that is known as miniature end plate potentials. Right? The other thing that will happen is that it will cause opening of potassium voltage gated channels, and these potassium voltage gated channels will cause repolarization, which will close the calcium voltage gated channels and prevent the release of neurotransmitters. So aminopyridine is a blocker of these potassium VGCs. Aminopyridine is a blocker of these potassium VGCs, and that is how it will help alleviate the symptoms of Lambert-Eaton myasthenic syndrome. Right? That is how the aminopyridine works in Lambert-Eaton myasthenic syndrome. At the motor end plate, what will happen? The acetylcholine will bind to the acetylcholine nicotinic receptor. It will bind to the nicotinic receptor. Okay, there are multiple types of nicotinic receptors. The one at neuromuscular junction is called as NM. Okay, it will open the ligand gated channel and it will cause the influx of sodium. This influx of sodium will also open sodium voltage gated channels. So these are produced. They are known as end plate potentials. These are examples of graded potentials. These graded potentials will in turn open the sodium VGCs and they will cause depolarization or action potential, and that action potential will then lead to excitation contraction, and that will lead to excitation contraction coupling, right? What is the fate of acetylcholine? Acetylcholine is degraded by acetylcholine esterase, and we use acetylcholine esterase inhibitors in case of myasthenia gravis. Then acetylcholine will degrade into acetate and choline. Choline is reuptaken by the presynaptic terminal and acetate is used by other molecules, that is sent to other pathways, right? Acetylcholine is broken down in this way. Okay. So muscle action potential versus nerve action potential is one difference. Okay, you can have a look at this if you want. Nerve is made for conduction, whereas muscle is not made for conduction, so its velocity is very less as compared to that of nerve. Okay. So there is something known as safety factor in neuromuscular transmission. What is safety factor in case of neuromuscular transmission? Safety factor is basically that the peak amplitude of the end plate potential, it should be very, very greater than the threshold potential for the action potential. What does that mean? It basically means that if a neurotransmitter is being released at the neuromuscular junction, end plate potential produced should at least be so strong that it goes over the threshold potential. Okay, so safety factor is equal to peak end plate potential divided by threshold potential, and for muscular contraction it should be always be equal to or greater than 1. If it falls less than 1, then you will have blockage of neuromuscular transmission. You will have blockage of neuromuscular transmission, and that will cause either some disease or that will cause anesthesia. That will cause anesthesia. Sorry, not anesthesia. That will be like a muscle relaxant. Alright, you will not be able to move your muscle. That will be like a paralytic. Okay. So safety factor is a very important concept that you have to understand. Say for example, this was the threshold that was required for an action potential. Let's say this was the threshold that was required. So end plate potentials that are produced, they will try to be above this. So this is the additional amplitude that is there. So the ratio of this peak end plate potential with the threshold is the safety factor, and it should always be greater than 1 for efficient neuromuscular transmission. So how is safety factor maintained? First of all, you have excess release of synaptic vesicles. Much more than required synaptic vesicles are released by the presynaptic terminal. Then you have junctional folds. They will increase the surface area, so they will also increase the safety factor. Then you have high density of sodium voltage gated channels. These sodium voltage gated channels are low threshold isoforms. So you have low threshold isoforms of sodium voltage gated channel in NMJ. And you have higher ACH single channel conductance with maturation of NMJ. What does this mean? That as a neuromuscular junction becomes older, the single channel conductance of an ACH receptor increases. That is, the ACH receptor allows more sodium to pass through a single channel as it becomes more mature. So there are 5 factors which contribute to a decent safety factor, and reduction in any of these 5 factors will cause a decrease in safety factor. And if it decreases below 1, then neuromuscular transmission becomes ineffective. For example, in case of Myasthenia Gravis, what happens? In case of Myasthenia Gravis, you do not have higher ACH single channel conductance. This acetylcholine channels, they become less. So because of that, safety factor decreases and neuromuscular transmission becomes ineffective. So that is everything that you have to see. Similarly, in case of botulism, there will be failure of release of synaptic vesicles. So all of these diseases that will affect the neuromuscular junction, they will reduce the safety factor. Okay. So next we are going to talk about drugs that will affect the NMJ. Okay. So when we talk about drugs affecting the neuromuscular junction, we're going to talk about two or three different kinds of drugs. First are drugs that will mimic or prolong the action of acetylcholine. There are drugs that will mimic or prolong the action of acetylcholine. They are known as depolarizing. They are depolarizing, and they are basically analogs that are very similar to acetylcholine. They will prolong the action of acetylcholine. So they can basically be used in case of your myasthenia gravis. They're basically used in case of myasthenia gravis. Okay, so drugs like methacholine, carbachol, and nicotine. These are all drugs that will mimic the action of acetylcholine. That is why acetylcholine receptors. That is why acetylcholine receptors are known as nicotinic. They are known as nicotinic acetylcholine receptors. They are known as nicotinic acetylcholine receptors. All right. That's the first thing. That's the first thing that you have to know here. Okay. Methacholine, carbachol, and nicotine. All of these are drugs that will mimic the action of ACH. These are the ones that will mimic. Now, what are the drugs that will prolong the action of acetylcholine? They are basically acetylcholine esterase inhibitors. Now, there are reversible inhibitors and there are irreversible inhibitors. There are reversible inhibitors and there are irreversible inhibitors. Now, reversible inhibitors are used in case of myasthenia gravis. They are used to treat myasthenia gravis. Neostigmine, pyridostigmine, all of these are used in case of myasthenia gravis because they will block acetylcholine esterase and they will prolong the action of acetylcholine and they will block the action for several hours, but then they will get deactivated. But there are some drugs which are irreversible, like diisopropyl fluorophosphate, right? This is basically nerve gas. This is basically nerve gas and its action takes place for weeks. It will block acetylcholine esterase for weeks. So then it will cause paralysis. It will cause paralysis. Why? Because then acetylcholine will never get dissociated and then lots of acetylcholine will get accumulated in the NMJ, causing excessive spastic paralysis of that muscle, right? So there is a ACH acetylcholine esterase inhibitors can be useful also and they can be poisonous also. So excess of everything is bad. So that's the principle that is going to come into play here. That's the principle that is going to come into play here. All right. Then you have neuromuscular transmission blockers. You have neuromuscular transmission blockers. So they are competitive as well as depolarizing blockers. Competitive blockers are non-depolarizing. What they will do is basically they will bind to the ACH sites and they will block the binding of ACH. They will simply block the binding of ACH. They will not do anything else. Alright. So they will block the binding of ACH. So they can be ultra short, they can be intermediate, or they can be long. And example is curariform drugs. Example is curariform drugs. Example is curare. Example of this is curare. All of these drugs will have curare in their words. For example, pancuronium. Pancuronium. It is a curariform drug. So it has CUR. So this is the identification of curariform drugs. Okay, curare was a molecule that was used by hunters and they put curare on the tip of their arrowheads while hunting animals. So these are neuromuscular transmission blockers, non-depolarizing type. Then depolarizing ones, they will bind to acetylcholine receptor and they will produce persistent depolarization, but they are not broken down by acetylcholine esterase. They are not broken down by acetylcholine esterase. Okay, for example, succinylcholine. They are of ultra short duration. Succinylcholine is an ultra short duration depolarizing blocker. Succinylcholine. Alright. It is a depolarizing type of blocker. Okay, it is similar to acetylcholine. But because acetylcholine also has a choline group, this also has a choline group. But the difference is that it is not broken down by acetylcholine esterase. So it will produce persistent depolarization first and then there will be sodium channel inactivation. So it will produce spastic paralysis, but again it will not be degraded by acetylcholine esterase. So it will put the muscle into paralysis and it will cause blockage of neuromuscular transmission. So now you know that there are drugs that mimic the action of ACH, there are drugs that prolong the action of ACH, then there are nerve muscle transmission blockers. Alright. Now there are two more conditions that you should know about, that is botulism and tetanus toxin. How do they work? Botulism, what it does is there is a Clostridium toxin of that disrupts the molecular machinery of fusion of the ACH vesicles with the presynaptic membrane of nerve terminal. They will basically target synaptosome associated proteins. There is a botulinum toxin A and E and there is a botulinum toxin B. The botulinum toxin A and E, they will cleave SNAP25. SNAP25 is the synaptosome associated protein. Alright, and botulinum B will cleave synaptobrevin, which is a vesicle associated membrane protein. There is something known as vesicle associated membrane protein, and there is something known as TAMP. Okay, there is VAMP and there is TAMP, right? Anyways, not going into the details of this, but what you should know is that botulinum toxin, it will basically affect synaptosome associated protein, or vesicle associated protein. It can affect anything, right? And if it affects these proteins, it will prevent the fusion of ACH vesicles, and it will ultimately prevent the release of ACH vesicles, and it will cause descending flaccid paralysis. What is descending? That is, it begins in the upper body and it moves to the lower body. What is the meaning of flaccid paralysis? Flaccid paralysis means that the muscle is in a non-contracted state, because the muscle cannot contract because ACH vesicles are not being released, and it is affecting synaptosome associated protein or synaptobrevin. Botulinum toxins A and E, they will cleave SNAP25. Botulinum B will cleave synaptobrevin, which is a vesicle associated membrane protein, and they will prevent the fusion of ACH vesicles with the presynaptic membrane. Alright, these proteins, these synaptosome associated proteins, synaptobrevin, these are required for the fusion of ACH vesicles with the presynaptic membrane. Okay, synaptosome associated proteins, vesicle associated membrane proteins, they are important for exocytosis. And if they are blocked, then exocytosis will not occur. Alright, do you understand this much? Now I move on to tetanus toxin. We'll move on to tetanus toxin. Tetanus toxin, it binds irreversibly to the presynaptic membrane. Then it undergoes retrograde axonal transport. So see what happens. So this was your synaptic cleft. Here you had tetanus toxin. It gets bind to the presynaptic membrane, and then it undergoes retrograde transport. Retrograde transport is from the axon to the dendrite, where it reaches the cell body of the motor neuron. Here it is picked up by terminals of the presynaptic inhibitory neurons. It is picked up by the terminals of the presynaptic inhibitory neurons. It is picked up by their terminals. This toxin, it will attach to gangliosides in these terminals, and it will block the release of glycine and GABA from here. Because of that, this inhibitory neuron is not able to play its function. Okay, so that will cause increase in the motor neuron activity of this motor neuron, and that will cause spastic paralysis, lockjaw of masseter. Okay, tetanus toxin, I will explain to you in a very beautiful way, because it's a very beautiful understanding. It explains you two concepts. It explains you Renshaw cell inhibition also, and you will understand tetanus toxin also. Okay, see what happens. First, you have to understand what is a Renshaw cell. Renshaw cell is an inhibitory interneuron. So how does Renshaw cell exactly work? So basically, Renshaw cell, it has its dendrites from here. It takes its dendrites from here, right? We'll draw this in different colors. You have to understand how a Renshaw cell works to understand how tetanus toxin is working. This one I'm drawing in white, this is a motor neuron. Okay, this is important for your motor physiology also. So try and understand this. This is a motor neuron. Alright. So when it receives an impulse to fire from a higher motor neuron, right, it will fire. It gives a collateral. It generally gives a collateral here to another cell, that is known as your Renshaw cell. Okay, that is known as your Renshaw cell. It is an interneuron. and then it will give an axon that will cause inhibition here, that will cause inhibition of the motor neuron. So this is a feedback loop. This is a feedback loop. All right. The motor neuron, when it excites the muscle, it gives off a collateral that gives afferent to the Renshaw cell, which causes release of glycine and GABA, causing inhibition of the motor neuron. So this is a feedback loop. Right? You understand this? Now what happens in case of release of tetanus toxin? Tetanus toxin, it will come here. And it will bind to the presynaptic terminal of motor neuron. Then it will undergo retrograde transport here, and tetanus toxin arrives here. Tetanus toxin will then bind to the gangliosides here at the Renshaw cell. It will bind to the gangliosides here, and it will block the release of glycine and GABA. Now that will disrupt the inhibition, that will disrupt the feedback inhibition. And since that inhibition is disrupted, there is excess firing here. So that is called as spastic paralysis. So that is how the tetanus toxin affects the Renshaw cell inhibition and causes spastic paralysis. Okay. And that was why, and most commonly it affects your masseter. So that is known as lockjaw of masseter. Alright. So we are done with all the questions that were there in the physiology essentials that I had shared with you guys. So you guys can see that I have covered these many questions. And guys, I assure you that if you do this much, that's all you need to do for nerve muscle. Alright. I will cover some extra topics just for the sake of completion. So see, we have covered these 10 questions for nerve muscle: A, B, C, D, E, F, G, H, I, J. You should be able to write 10 markers for all of these. You should be able to answer most of the questions in your Viva. And I am assuming you have studied nerve muscle once before also for your internals. And this should complete your Prof preparation for nerve muscle physiology. So I will cover some extra topics just in case they are asked in your Viva, or they come as a 2-marker question or a short answer question, right? Because sometimes these are asked even in MCQs. So first of all, you have to know about the classification of nerve fibers. Very important. There is a classification that is known as the Erlanger and Gasser classification, and there is also Lloyd's classification. Alright. This 1A, 1B, 2 and 3, this is the Lloyd classification. The rest is Erlanger-Gasser classification, right? जो alphabets वाली है वो Erlanger-Gasser है, alphabet वाली, और जो number वाली है वो Lloyd classification है. ठीक है? इसमें what you should know is that there is A fibers, B fibers and C fibers, and only the C type of fibers are unmyelinated. Rest all are myelinated. ठीक है? A fibers and B fibers, they are myelinated, C are unmyelinated. A are further divided into A alpha, A beta, A gamma and A delta. A alpha fibers, remember, are for proprioception and somatic motor. A beta are for touch, pressure and motor. A gamma are intrafusal fibers, that is they are motor fibers of muscle spindle. Please remember, A gamma fibers are motor fibers of muscle spindles. And A delta fibers are pain, cold and touch. Very important, pain fibers. They are responsible for paresthesia, right? The diameter decreases as you go down, and these are unmyelinated. So these are the slowest and these are the fastest. A alpha are fastest, then A beta, then A gamma, A delta, B and C, right? Then the Lloyd classification is only for the sensory fibers. It is only for the sensory fibers. So 1A and 1B are for proprioception, then 2 are for the touch and pressure sensations, and 3 are for pain sensations. 4 are the dorsal root ganglion fibers, which are for pain and temperature. Then B type of fibers are preganglionic autonomic, and the C unmyelinated fibers are motor fibers. They are pain fibers. They are also pain fibers, and they generally carry which kind of pain? Visceral pain, right? Now, differentiation between type 1 and type 2 fibers. Type 1 are slow twitch fibers and type 2 are fast twitch fibers. So they have slow tension development. These have fast. They have a slow myosin ATPase. They have a fast myosin ATPase. They have very well developed sarcoplasmic reticulum. Why? Because sarcoplasmic reticulum is required for instant increase in calcium and muscle contraction. So they have well developed sarcoplasmic reticulum. And their rate of relaxation is fast. They have a faster SERCA1, that is SERCA1. They have SERCA1, which is faster. SERCA1 is present in type 2 fibers because it is faster, and that helps in quick relaxation. Whereas SERCA2 is slower and it is present in type 1 fibers. They have oxidative fibers. They have mixed fibers in type 2A and glycolytic fibers in type 2B. Glycolytic capacity is high in case of type 2 and oxidative capacity is high in case of type 1, right? Mitochondrial volume is also high in type 1, capillary density is also high in type 1, and myoglobin levels are also high in type 1. Diameter is moderate for type 1 fibers, small for type 2A, and large for type 2B. These are fatigue resistant, these are also fatigue resistant, and these are fatigue susceptible. So this table should be on your tips, and you might also get to differentiate this in your examinations, and you might get this as a question here, right? Classification of peripheral nerve injuries. There is a grade 1. So there is a Seddon classification and there is a Sunderland classification. Seddon classification is very simple. Neuropraxia, axonotmesis and neurotomesis, right? That's the here the Seddon classification versus the Sunderland classification, right? Neuropraxia, axonotmesis and neurotomesis. ठीक है, पहले just try to understand from the Seddon classification. Let's not focus on the Sunderland classification, right? Let's remove this. See here, neuropraxia basically means that there is only some demyelination. This is basically physiological damage, right? There is no anatomical damage. There is only physiological damage. There is physiological damage. There is no anatomical damage per se, right? But when you move to axonotmesis, the axon is damaged. Here, only the axon is damaged, but the nerve is preserved. So that is why we say axonotmesis. And when we say neurotomesis, it means that the nerve is damaged. There is complete nerve transaction. Alright. So that is the basis of the Seddon's classification. Sunderland classification, it is more complex. It divides the axonotmesis into 3 grades. Grade 2, grade 3 and grade 4. Neuropraxia is grade 1 injury and neurotomesis is grade 5 injury. Alright. Here you will have focal segmental demyelination. In this case, you will have the damage of axon without damaging endoneurium. Here endoneurium is damaged but perineurium is intact. Here perineurium is also damaged but epineurium is intact. And here there is a complete nerve transaction. So you will be able to see this here. That what is grade 1 injury? Grade 1 injury is neuropraxia. There is only some demyelination here. Rest everything is intact. In grade 2 axonotmesis, the axon is also damaged. But the endoneurium is intact. In grade 3, the endoneurium also gets damaged. In grade 4, the damage is up till the perineurium, but the epineurium is preserved. In grade 5, there is complete nerve injury. Okay? Besides that, you can also read about Wallerian degeneration and Wallerian regeneration. Okay? That is another topic that is sometimes asked in a viva. Okay? So that is about the theoretical aspect here so far. Okay? So take 5 minutes, revise the entire theory, just skip through the entire lecture and tell me if there are any doubts. If there are any doubts you have so far. First tell me any doubts that you have, then we'll move to the MCQs. Or any feedback you have regarding the theory session? Okay, so I tried to complete the entire nerve muscle in 3 hours. Okay, so otherwise it's a huge topic. It's an entire unit of your guidance. So again, this is an attempt to help you with your prof examinations. Hello. We've done the first question. Can you answer this question, guys? Initiation of impulse, where does it start? So I told you that this is a previous year question of Nimhan's, and the impulse, it begins in the axon hillock or the initial segment of the axon, right? Second question, question number 2. Neuropraxia is a type of. This question is a recent question. It was asked in the JIPMER examination in 2014. Any answers, guys? I just told you this in the lecture. I was waiting for a response from your end. So essentially, neuropraxia, it is a type of physiological conduction block. I just told you neuropraxia, you do not have any anatomical damage. You don't have any anatomical damage. So there is only some focal demyelination. So there is no axon disruption. There is no disruption of endoneuron. The nerve trunk is also continuous. There is no damage to the axon, endoneuron, or the nerve trunk. There is only some demyelination. There is only some demyelination. So there is a physiological. So I'll call it a physiological conduction block, right? Let's move to question number 3. If one of your nerves is compressed and this leads to paresthesia for some time, the type of nerve fiber affected is probably, okay, so you tell me the type of nerve fiber that is affected. Right. So seems like you guys don't know the answer to this one as well. It's a very old previous year question. So the answer to this is A delta fibers. So A delta fibers are the ones that I told you, that the A delta fibers ones that are involved in paresthesia. So that's why I taught those last miscellaneous topics, because they often figure out in MCQs, although they're not very much asked in your prof examinations, but they're often asked in your vivas and your MCQs. So do go through these last topics very well. All right. There is also a table regarding various susceptibilities. A fibers are susceptible to hypoxia, pressure, or anesthesia, right? So A fibers, B fibers, and C fibers have different types of susceptibilities. So even you should read those out, right? Right. So somebody has answered. Deepika has now answered for this question number 3. The answer is A delta fibers. Very good. Question number 4. Let's do this one. This is a calculation one. Deepika has calculated this question as 30 meters per second. That's absolutely the correct answer. So you can see that the latent period, this is the time that is there for conduction. So you're directly stimulating the axon. So there is no other delay. The latent period here will refer to the time that is taken by the nerve to conduct, right? So 4.5 centimeters is the distance traveled, all right? And the latent period is 1.5 meter per second. So now it's a very simple formula that speed is equal to distance upon time. So just apply that and you should be able to this 0.5 milliseconds. This is 1.5 milliseconds. So 4.5 into 10 raised to the power minus 2 for centimeter, and 1.5 into 10 raised to the power minus 3 for milliseconds. So 10 will come above. This is 3 milliseconds is your correct answer. So that's there for question number 4. I hope you are able to solve this. Just have another look at it. And if you want to take a screenshot of the question, you can do that. Question number 5. So the question says, find out the chronaxy and the rheobase. So this is not something I've taught you in this lecture, but I am assuming that you know a little bit about chronaxy and rheobase. So try and attempt this question. So Jeevan Rathod has answered this question. Chronaxy is 0.22 milliseconds and rheobase is 0.68 volts. So just have a look at this question very carefully. So the rheobase is basically nothing but the but the minimum that is required, right? What is the minimum that is required here? So there are two things. One is the chronaxy and one is the Rio base. So do we have any other answers also? So it is the minimum strength of the stimulus that must be applied, right? Rio base is there must be a minimum strength of the stimulus that is applied here, right? There must be a minimum strength of the stimulus that is to be applied, right? So that minimum strength of the stimulus, that is this much. That is this much. Okay. So if you look here, this is 0.1, this is 0.2, this is 0.3. So this is almost 0.35 volt. That is going to be the Rio base. That is going to be the Rio base. Now chronaxy is the time of stimulus required for twice of the Rio base. For the twice of the Rio base. Alright. So again, so Rio base is 0.35 volt, and if you go to 0.7 volt, so this is 0.5, and if you go here, this is 0.7 volt, and corresponding to that you have to see the time interval, that is somewhere around 0.22 milliseconds, between 0.2 and 0.3, so it's 0.22 milliseconds. So the answer to your fifth question should have been option A. The answer should have been option A, right? Jeevan Rathod, you said that the answer is D. It's not D. It should be answer should be option A for question number 5. Am I doing something wrong? You can let me know. If I'm doing something wrong, you can let me know. Jeevan, are you clear with this question? Okay, okay. I think Jeevan को समझ आ गया. Let's go to question number 6. Again, let's see if you can answer this correct. ठीक है? I think ये second last question है. So Deepika has answered this question. The answer that has been given by Deepika is D. Alright. So now I'll tell you the major pitfall in this question. The diagram shows the force velocity relationship, right? It's the force and contraction velocity relationship of the skeletal muscle, right? So when the force is zero, so what is the force here? This force here, this is the afterload, right? This is the afterload. So you can see here when the afterload is zero, the contraction velocity of V1 is maximum, V2 is lesser, and V3 is the minimum, right? So if you change the muscle mass or the frequency of muscle contraction or the recruitment of motor units, you will be able to have greater afterload. You will be able to increase the force that can be lifted. You will be able to increase the force that can be lifted. But only myosin ATPase activity is the one. The answer will be C. Only myosin ATPase activity is the one that can change the contraction velocity when the force is zero or the afterload is zero. All right. So that's for question number 6. Question number 7. Action of calmodulin is. Action of calmodulin is. So this is a multi-answer correct. This is a multi-answer correct question. You should see that there are two options that make sense. All right. It is a multi-answer correct question. You all are right. Deepika and Jaswinder, you are right that A is correct. That action of calmodulin is dependent on calcium. Calcium dependent. Right. A and D, no, that's wrong. A and D is wrong. Okay, Deepika is right. It is B. Through calmodulin dependent kinases. So there is a myosin light chain kinase, and that is dependent on calmodulin. So calcium calmodulin myosin light chain kinase is the active form of the myosin light chain kinase. So the action of calmodulin is calcium dependent, and it works through calmodulin dependent kinases, which are myosin light chain kinases, which are dependent on calmodulin. Here. So are there any other doubts for this lecture? That's the end of this lecture. Thank you so much for watching. So my plus referral code is Chetanya10. So you can use this referral code to get a 10% off on your plus subscriptions. Use this referral code to get a 10% off on any subscription that you take, whether it be a plus subscription or an iconic subscription, or whether if it's a subscription for your brother or sister for NEET, UG or UPSC or anything. Do hit the bell icon for notifications, subscribe to the channel Let's Crack NEET PG, hit the like button on this video, and do drop in the comments what other videos you want from our channel. And you can subscribe to us on Telegram. The Telegram link for the common channel for this YouTube channel is t.me/ go to Let's Crack NEET PG. Let's Crack NEET PG. Go and subscribe to this channel. Okay, and here you will have updates from the entire team of this channel. Go ahead and subscribe to this channel, right? Thank you so much. Take care. I'll be ending the stream now. Okay, thank you, Deepika. Thank you, Jeevan. Thank you for participating.